Hi, I'm Casey Broecker. So this summer I worked with Yong Chen here at Stony Brook on the Hudson River Biological Monitoring Program. And basically my role was to determine kind of how the Hudson River program was kind of being used for policy related changes within the system. So just kind of some background on the project. In 1963, there was a lot of citizen and environmentalist concern over the building of nuclear power plants along the Hudson. And when Unit 1 of the Cornwall pump storage was being approved, they did not need approval for their once-through cooling systems, which was then later used for Unit 2 and Unit 3. But then in 1969, the National Environmental Policy Act was passed, which was basically, once the act was implemented in 1971, the Atomic Energy Commission was then implemented, which required non-nucleotide effects to also be considered when constructing the power plants. It was determined that the cooling systems could not really be modified at the time, back in the 70s, because the technology wasn't really there to do so. Also in 1971, the Clean Water Act was passed. So this kind of meant that there was a need to limit the environmental impact, which, like I said before, the cooling systems were not actually really a part of that until later on. This also kind of pushed for that to be changed. Sorry. Then in 1974, the Hudson River Biological Monitoring Program was basically born. The first survey was conducted in 1973. And then by 1981, there was the Hudson River Settlement Agreement, which basically said that they had to, that the Storm King project had to be abandoned on the river. They basically said, no, you can't build any more power plants except for the ones that have already been approved. So any new ones were a no-go. And they also had to provide 10 years of funding with $2 million for a biological monitoring program. And at the time, this was the most ambitious river survey in the entire United States. And we have over 40 years of data from this survey, because the survey is still currently going on. So basically the objectives of my project were to basically see over the course of the entire 40 years, how has policy and infrastructure kind of changed, as well as then if the data is or is not being used, how is that, why is happening? So I did three things. I conducted a literature review to kind of get more of a background on the overall project, kind of see what was happening, how data was being collected, what kind of projects were being used, and then I would also review management organization documents to kind of determine the work that they have done and the work that they're currently doing. I also reviewed interviews that have been previously conducted by other lab mates, and that was more to get an understanding of both sides kind of the project and hear what people who have been involved with the project for however many years, how they learned about the project, how they used the project, what they thought needed to be continued. And finally, I sent out a survey to the New York DEC, basically to determine what they think that Stony Brook could be doing better as far as data availability. So as far as what I actually found within the literature review, one of the main organizations that's doing work on the Hudson is called Riverkeeper. They are a nonprofit organization. But there are two projects that most closely relate to the biological monitoring program, which both involve the power plants. So Indian Point was obviously the big one. That was kind of the one that really got people stirring. And the power plant has been shut down now for 2 years. It shut down in August of 2021. And the plant produced 2,060 megawatts of power, which they were basically able to replace. But Riverkeeper's goal within that was to try to find clean and renewable energy to replace it so that they weren't still running into the same problems. They were also instrumental in making sure that the employees that were working at the plant did not just completely get cut and laid off. So they were also, they tried to keep the people on for the decommissioning process. The other power plant, the other power plant campaign that they also had, pretty much the same thing, making sure there's safe decommissioning, pushing for them to modernize their once-through cooling systems, and eventually pushing for them to get shut down. They have other projects which are important, but don't necessarily relate entirely to the Hudson project. They have the sewage overflow campaign, which basically is trying to push for better source controls, as well as possibly using the sewage for crops and things within agriculture, instead of just allowing it to dump straight into the Hudson. Contamination sites. So, fun fact, the Hudson is one of the biggest Superfund sites in the United States. Wow. Yeah. So, basically, General Energy was dumping a lot of PCBs into the Hudson, which naturally became a problem, which I will talk about later with striped bass. Pretty much, they were dumping 1.3 million tons over their course of operation. And Riverkeeper is kind of pushing for them to clean out the system, which they are currently doing. They are currently dredging for PCBs. And then finally, there's the Polluters campaign, which basically is a citizen-run operation. The citizens notice things that are not up to like code. They report it back to the DEC, and then the DEC kind of does whatever they do with it. Oh boy. So there was also a bunch of things that the DEC published. The main one, they created the Hudson Estuary Program, basically saying that the Hudson is in fact an estuary. We need to treat it as such. There are tides. It is not just a river. They produced an action plan, which is basically pushing for a more resilient Hudson, as well as environmental justice. So making sure that people are living safely with their environment, not just exploiting it. So as far as the interviews, this is kind of where I got most of my information, which I'm aware does not seem like that. But I focused on 3 interviews primarily with Fran Dunwell from the DEC. She worked there for many years and was involved in the project in many different ways. She was the Hudson River coordinator. And basically the biggest changes that she saw were within fisheries. So within her time there, the American shad fishery shut down, as well as the Atlantic sturgeon. Pretty much the data showed that there was not really a population to support those fisheries anymore. And striped bass also could not be fished for because they're full of PCBs. Her suggestion is that we need more permanent funding, because right now we're kind of working on a year-to-year basis, and however many years of funding we could get, we just work with that. The next person that I focused on is John Lipscomb, who is a patrol boat captain for Riverkeeper. And he was more knowledgeable on this in terms of like working with management teams rather than as a scientist. A lot of people aren't just focused on the fish, they're also really worried about the water quality because not everybody on the Hudson fishes. Riverkeeper's goal is to regulate the Hudson for the purpose of the Hudson, not for people. And basically we need to start asking ourselves, why are the fish not coming back? And then finally, Elise Zoli, I think is how you pronounce it. She was a lawyer for the management, for the energy companies, and basically by the time that she was hired on, there was still no answer as to what kind of impact the cooling systems were really having. So she brought on 3 scientists who basically conducted their own analysis and said, you know, this actually doesn't have as much of an impact as we thought it did. And one of these scientists was very adamant about the fact prior, saying, no, once through cooling, bad, no go. And they all came to the same conclusion, and they realized that good governance is really kind of what was driving populations. Really quick with the survey, pretty much the biggest takeaway is how to make it more accessible. There's a lot that I could get into with that. Basically, make the data accessible, publicize it, and possibly give a searchable database. So the main takeaways from my project overall is that the Hudson River Biological Monitoring Program can be used as a model for other estuaries along the Atlantic coast. Many of these species don't just reside in the Hudson, they exist outside of it. So other states that also have these species can look at our data and basically kind of determine how populations are doing, especially if they're trying to manage their own populations without any sort of long-term survey like we have. There are future power projects that are currently going on. There's talks of putting a power cable straight through the Hudson because offshore wind is a big deal. And I am about to be cut off, but we need more money. Yay, money. We need some sort of endowment, and data needs to be accessible. All my citations.
Okay, 3 minutes.
I'm so sorry. Yeah, Chris.
I don't have a general question, but in your opinion, what's the most pressing issue facing the Hudson River estuary today?
I mean, truth be told, the Hudson isn't necessarily the cleanest, but overall, I mean, there's something that is causing like the shad populations to not rebound. John Lipscomb that I talked about, he basically was saying like, yeah, we have a lot of juveniles of them, but like, we're the adults. So not really entirely sure what's happening with that, but there's something that's saying, yeah. I don't know if that really answered your question, but
I think that's it. Thank you.
All right, hi everyone. I'm Travis. I'm going to be talking about STEM belonging and the cure for student attrition. I want to get right into it because there's a lot to talk about. I promise this has to do with marine science, even if it doesn't sound like it. So what am I talking about?
click anywhere on the screen. There you
There
go.
we
Now it works.
So what is a CURE? CURE stands for curricula-based or course-based undergraduate research experience. And that is exactly what it sounds like. If you've ever taken like an early, especially a first or second year biology lab, you know that it's just following a set of pre-done instructions and you're reaching a set conclusion. This is the opposite of that. It is genuine research where there is no set conclusion and students are involved in designing and conducting the experiments themselves. Benefits of the CURE include improved critical thinking and the development of technical skills, obviously, because this is genuine research. You have no set guide rails. You're not following directions in that sense. It also improves your teamwork because you're not doing this by yourself. Of course, it's big, challenging work and you need a full team to do it. And it's good grad school and workforce prep because, again, it's genuine research. It's a more authentic experience of what you're going to be doing after your undergraduate career. So what are we actually curing? Not every student who enrolls in college graduates. This might be a surprise to you, maybe not. But one study at least suggests that nearly half of all students who enroll in a STEM degree leave STEM behind entirely within 6 years, which we're coming up on that soon, so we'll see. Cures promote STEM motivation and persistence, which means that they improve retention, right? It makes sense. If you're involved in more genuine work and you know you like it, then you're going to want to stick around. So, but why does it work? Well, like I said, active and experiential learning. If you're more involved in something, you're not just being told what to do. You feel more of as though you're more of a part of it. Authentic assessment. What this basically refers to is practical advice from your professors, rather than just you did this right, you did this wrong, you did this right, you did this wrong. It's you learned this. Here's how you can apply it to this scenario, this scenario, this scenario. It's more practical and it's more of a dialogue with the student. Peer-to-peer scaffolding, which basically refers to the idea that you're going to have a mentor who is your peer, a more experienced peer. This was the first of my responsibilities with this project. I was the graduate TA in a lab class, and I was the mentor to a specific group of undergraduates. And this is beneficial because while a good professor is a great thing, it's also very valuable in a very different way to have someone who's closer in age to you, who has more recently lived the experience that you are now living. And then sensor, I'm not going to read the acronym, I don't remember. Basically, all this means is that you're tying in the issues that you're dealing with real world civic issues. So if you're talking about contaminants, for example, you're going to tie that into legislation about landfills, something like that. So for the context of this project, the CURE class is SUS 352, conduct and communicate a research project in ecotoxicology. It just rolls off the tongue. We're going to call it Dr. Cochran's Worm Lab. And basically, Dr. Cochran built this lab to include all of those previous components of the CURE that we talked about. And like I said, I was a peer mentor. But normally it's designed for second, third, and fourth year students. I'll come back to that in a moment. So the real driving force behind this project, one of the two, is the CURE Plus. So in addition to cures, another function of cures that could help improve retention is the idea of social belonging. This makes sense, right? If you feel like you belong somewhere, you're going to want to stay around. If you don't feel like you belong, you're going to want to leave. So Cure Plus was designed to combine the benefit of social belonging with the cure via a belonging intervention, which took the form of interviews. You can see here my 6 setup, very high tech. And what these interviews consisted of were questions that forced students to reflect on their own sense of belonging. And this kind of positive affirmation creates a sort of feedback loop in their mind where saying is believing. If you say, I belong because, you're going to believe it more. So going back to the idea of what are we curing, there's little data on the effects of cures on first-year students. And so this whole, as I said, the Worm Lab was normally designed for second, third, and fourth-year students. So for this project, there were 2 sections, a Cure and a Cure Plus, both consisted entirely of first-year students. So if first-year students participate in these courses, how will it affect their GPA, their college and STEM fidelity, and their feeling of belonging? All right, so I've said what we're doing. Now, how do we actually do that? Because belonging is an abstract thing, right? At least I would think so. It's just you either feel it or you don't. Fortunately, this project was building off of a previous study that broke down how you can measure belonging with a 25 prompt survey. So you could be at the end of the semester, the students in both sections were given this 25 prompt survey, and they had a 1 to 5 scale from strongly disagree to strongly agree. And the 25 prompts were broken up unsatisfactorily into 5 categories. It was not 5 prompts per category. I don't know why. 5 categories. Social belonging. I feel like I belong in STEM. Self-explanatory. Ability belonging. I'm not sure that I'm cut out for STEM. STEM identification. It is important to me that I'm good at STEM. STEM self-efficacy. How much do you complete homework assignments by yourself? And intention to persist in STEM. I could see myself going into a career related to STEM. So after the spring semester and this survey was all done, I had the joyous task of crunching all the numbers. I'm not a statistician. I don't know how I got that job, but I did. So what I ended up finding was that there was a very high sense of belonging in both the Cure and Cure Plus groups. You can see here that the average score for 4 of the 5 categories in both sections was about a 4, which, considering 5 is the strongest sense of belonging and 1 is the strongest sense of not belonging, 4 is pretty good to be so consistent across the 4 categories. And then only in the ability belonging category was the average score for both groups 3. And 3 is not a bad score, it's a middle of the road score. And considering that these are first year students, if you'd like a refresher, ability belonging, I'm not sure that I'm cut out for STEM. I'm a grad student. I doubt my own abilities plenty. So it's understandable that first year students would be skeptical of their own capabilities. But what you might also notice with this lovely chart is there is no difference between the cure and cure plus. If you recall, the idea was to combine the belonging intervention with the cure, and that would make it even better, right? So we should have seen, if that was correct, that the light gray bars would have been way higher than the dark gray bars. But that didn't end up being the case. There was no statistically significant difference across any of the categories for the cure and cure plus groups, which suggests that basically the cure, the normal cure, is powerful enough that it induces enough of a sense of belonging in STEM that you don't need that extra intervention. There could still be a purpose to having the intervention, but I'll get to that in a moment. So why does this matter? Academic outcomes. I'll note first and foremost that these are not statistically significant results, but if you look at this table, the trend is in the right direction. The cure and cure plus students were more likely, had on average a higher GPA, were less likely to leave STEM, were less likely to leave school, and were less likely to receive an academic suspension. Again, not statistically significant, but this is the trending in the right direction, what we want to see. So going back to that belonging intervention, a final component of this project is trying to ascertain how belonging might manifest, right? Because if you can identify what belonging looks or sounds like, then you can modify the cure class or any class to incorporate those elements more to better support that feeling of belonging. So I'm going to hopefully see if this works, play a clip.
We might be able to get it to work.
All right. I mean, if not, I have the quotes. Should I just play the quotes?
Sure.
Should I just? Okay. That's a shame because this was a visual project, but look at this. I'm so prepared. Go. Okay.
Girl, yeah.
Slideshow. Slide.
Maybe a bit more. Yeah.
Okay. So these three quotes are all in response to the question, how do you belong in STEM? So the first quote here talks about feeling like the student belongs in STEM as opposed to going to art school. They thought they wanted to go to art school, and then they realized that, no, I enjoy doing the scientific work more. So that's just a joy of the work. The work makes you feel like you belong. The second student was talking about feeling a need to act as a representation for historically underrepresented groups in STEM. So that's a sense of justice of sorts, that it's not about necessarily the work itself, it's about being a good example within the work. And the final student said that he didn't really feel like he belonged, but he was working to stay in STEM and trying to earn that feeling of belonging. Now, you might say that that's not feeling like you belong. This is all very subjective. But what it suggests to me, anyway, is that there's a craving to belong, and that craving is in itself a form of belonging. And then another thing I did with these interviews, after myself and two undergraduate TAs filmed the interviews, we transcribed them, and then in the aftermath, I used those transcripts to create this lovely word cloud graphic. And I just want to highlight two words that have occurred quite frequently: fun and cool. As someone who has taken Bio 204 and 205, I can safely say I never used either of those words to describe either of those classes. So this has to count for something. So conclusions for now. So Cure and Cure Plus both seem to improve academic performance, or at least they're trending in the right direction. They both maybe improve STEM belonging, certainly looks that way. There's no difference between Cure and Cure Plus, and that's really the big finding for this first year of this project, that there is no difference between the Cure and Cure Plus. And the interviews could yield interesting insights, like I said, but this is the first of a 3-year project. So this first cohort of students will be followed for the next couple of years to see what happens to them. So when I talk about how belonging manifests, 2 years down the road, we could see, oh, this student said this in the interview and they dropped out, and this student said this and they stuck with it and they're doing really well. That could give us some insight into how belonging manifests and what you can focus on to improve that sense of belonging. So I just want to quickly thank SoMAS, the MCP program, Dr. Taylor for being my faculty mentor and guiding me through this, Dr. Cochran for creating the Worm Lab, and along with Dr. Taylor hiring me for this position. Alex and Christina, my undergrad TAs, the best helpers and friends a guy could ask for, and of course the Worm Lab students. They didn't know that they were part of this project, they were a joy to work with. Thank you.
Were there any like barriers to entry into the class?
None that I can talk about.
No, like,
Not like a barrier. There were certain criteria to the project. Can't talk about it.
Do people voluntarily choose if they are joining Fier versus Fier class?
No, that was just chance. It was just there was another graduate TA who was the TA for the Cure. I was the Cure Plus, and that's just how it happened.
What exactly was your control when you were comparing?
Oh,
academic?
you're talking about, yeah. This one? So,
Yeah.
yeah, so for this one, the control was just students who weren't in these classes. It was a group of about 200 students.
Okay,
So
Where-
compared to the 50 across the two Cure sections, it was 200 in the control. That were in
So-
normal lab classes.
okay, were they like biochem, marine science?
Yeah, I don't know the specifics of which course it was, but it was, what do you mean, like the Cure students?
No, the control.
Yeah, I'm not sure which classes were
Okay.
used for the control, but it was topical first year students.
Okay,
Is that it? We good?
sorry. Had to make it run awkward. So you said it's a 3-year project.
Yes.
Are they planning on doing another like Cure, like adding to the pool of students who have gone through Cure and Cure Plus? to
Yes.
Hopefully pull out the statistics a little bit
Yeah,
better?
to improve the sample size, yes.
Yeah.
It'll be another pair of grad students next year and the year after that doing a Cure and Cure Plus, and we'll get more data. But these are just the results of this first year of the project. Obviously, I'm not going to be involved in succeeding years. So.
Okay. All right, hello, everyone. Again, I'm Chris Eagler. The title of my project is Assessment of Piping Plover Population and Productivity Trends in New York State between 2001 and 2022. So brief introduction about myself and my internship. I'm a wildlife technician with the New York State Department of Environmental Conservation. We deal with all things wildlife, ranging from hunting regulations. We banned a ton of birds. But in the summertime, our main priority are these little guys here, the piping plovers. So because of that, I decided to focus my project on those little birds. So the purpose was to analyze these trends and hopefully achieve a deeper understanding of the status of piping plovers in New York State, and in doing so, providing valuable information to DEC biologists who lead conservation in New York State. Some background on the bird. It's a small tan shorebird found all across the east coast of North America. There are inland populations, but this is focusing on the Atlantic population. They winter down south, but their breeding range stretches from Virginia to parts of Canada. So in New York State, they typically arrive by late March. Pairs will begin nesting by late May, early June. Females will lay a nest of 4 eggs in discrete depressions in the sand, as you can see here. Very easy to miss if you're not looking for them. Incubation lasts about a month, and once the chicks hatch, they are vigorously protected by the adults until they gain the ability to fly or fledge at about 28 to 35 days old. And by early September, most broods have begun their journey back down south. So some threats that have affected plovers historically and are still today. Habitat loss is a big one. Coastal development, dredging, beach nourishment, all limit the amount of available habitat they can nest in, as you can see with some of those estates in West Hampton. Recreational activities, specifically off-road vehicles, are a big one. As you can see, they like to hang out in tire tracks, and that becomes more of a serious issue when there are chicks on the ground, as they don't have the ability to fly out of the way. And predators made abundant due to human activities. On Long Island, fox, raccoon, cats, crows, ravens are all posing threats to piping plover populations. So the species was originally listed as threatened under the Endangered Species Act in 1986 by the U.S. Fish and Wildlife Service. The initial recovery plan was implemented in 1988, but revised in 1996. The goal of this plan was to remove the species from the endangered species list. And to do that, they have to meet some certain criteria. The first one is an overall pair goal of 2,000 pairs on average for a 5-year span across the whole breeding range. It's broken up into 4 management units. The New York, New Jersey unit, the goal was 575 pairs. There's also an overall productivity goal, which is productivity is defined as fledges per pair. The goal across all regions is 1.5 fledges per pair over a 5-year period. So management is delegated to the states. So New York management is led by the Department of Environmental Conservation. And management tactics implemented here consist of symbolic string fencing around nesting areas, the restriction of off-road vehicles during breeding season, and installation of predator exclosures, as you can see that bird sitting behind one. So my methods, I essentially compiled pair and productivity data for all 135 sites in New York State over the last 21 years. I then broke down these sites into 3 regions: South Shore, North Shore, and Bays. And I further divided those South and North Shore sites into Western, Central, and Eastern units by town. After doing that, I was easily able to compare trends in pairs and productivity across different regions. And I also utilized regression analysis to study the relationship between productivity and growth of the following year. And using that relationship, I was able to determine the productivity necessary to maintain a stable population. So just getting into some results, composition by region remained pretty stable over time. As you can see, the South Shore provides habitat for by far the most plovers. In New York, around 69%. And the North Shore and Bays contribute a much smaller percent to the overall population. So across all sites, there is a statistically non-significant increasing trend over time. On average, there are 408 pairs per year. For reference, in 1989, 191 pairs were counted. By 2001, we had 321 pairs here. 2022 was actually our highest year with 559 pairs. And the difference between 2022 and 1989 represents 293% growth. And also for reference, our carrying capacity here is estimated to be between 411 and 450 pairs. So we're hovering right around there for quite some time now. So on the South Shore, across all sites, there was no significant trend. However, the Western unit on the South Shore, which is Queens County and Hempstead, did show a significant increasing trend over time, despite having the lowest productivity of all regions analyzed. In the Central and East region, there were no significant trends. However, I would keep an eye out on the Central region going forward. It contains a very unique site called Democrat Point. This is the western tip of Fier Island. In the winter of 2022, the Army Corps did a habitat enhancement project there. And since then, productivity for many shorebird species has been remarkable. And over the last 2 years, they've had the most pairs in the site's history. So you could expect to see some more growth in that central unit going forward. On the North Shore, no compelling trends here. This just shows that most North Shore plovers are found in the towns of Huntington, Smithtown, and Brookhaven in that central unit. And again, the composition remaining relatively stable over time, fluctuating from around 50 to 80 pairs. In Bays, again, no significant trends here, just fluctuating from around 45 to 85 pairs per year, again, representing a smaller contribution to the population. So here's population growth in table form. So it's important to understand that a growth of 1 would indicate a stable population. So, for example, comparing pairs in 2001 to 2022, there was a 74% growth across all sites. And that growth is pretty stark for all comparisons from 2001 to 2022. But on average, all sites in all regions have shown gradually growing population with values just above 1. So here's productivity for a stable population. Again, a growth of 1 indicates a stable population. So for all sites, a productivity of 1.08 would support a stable population. And just looking at average productivity over the last 21 years, average productivity for all sites and all regions greatly surpasses the productivity necessary for that stable growth. So that's a pretty promising sign that management has been effective here. So this study, along with multiple other studies, have shown that the productivity to maintain a stable population in our region is well below the recovery goal of 1.5. This sort of indicates that the one-size-fits-all recovery goal does not accurately represent regional life history discrepancies. There's some evidence to support latitudinal differences, as more northerly sites require higher productivity. to maintain stable population. And that could possibly be attributed to the longer migration, increased mortality during that. The gradually growing population, despite the lack of trends in productivity, indicates effective protection of nesting habitats in New York. And since our population is in pretty decent shape here, we could start drawing some inspiration from Massachusetts. They implemented a habitat conservation plan in 2016, which essentially loosens restrictions based on abundance of the previous year, while also requiring minimization or mitigation measures that benefit the total population. So this could allow off-road driving during the breeding season with trained escorts, while also encouraging predator management and habitat enhancement at other sites to overall cause a net benefit. So going forward, obviously it's important to continue thorough monitoring to ensure accurate pair and productivity data. In my experience, this could be done by more banding. Having bands on birds makes it much easier to distinguish between pairs, and that could lead to much more accurate productivity data. Furthermore, biologists could use the stable population productivity rates identified here as sort of a realistic reference point of where the population's at, and sort of give them an idea of what it's going to look like in the following year. And finally, since our population here is in good shape, we could start developing a habitat conservation plan to increase pair numbers and productivity while also enhancing recreational and economic opportunities near the coast. I'm sure many of you know that a lot of people, for lack of a better term, hate piping plovers. They associate them with beach closures, restricting access to good fishing spots and whatnot, and that benefits no one. So sort of making that compromise and benefiting the overall population could increase public support for the conservation of plovers, again, leading to a net benefit going forward. I just want to say thank you to Dr. Carl Safina, my faculty advisor, and my DEC mentors, wildlife biologist Casey Pendergast and Chip Hamilton, who really gave me a lot of advice and knowledge throughout this whole process. Thank you.
Not sure if you said this, but does the recovery values, like in the bays, it was lower, does that have anything to do with like how much of that habitat was available to them?
Yeah, bays, it's not their optimal habitat. Again, the South Shore is their optimal habitat. So as far as bays go, I think it's just the lack of available habitat there. That's leading to that low composition.
Okay.
It's not like banding data. How often do the piping plovers come back to the very similar areas that they've bred in the past year? Do they change?
I don't know the exact data on that, but I can tell you that they do move around. I actually identified a band number in Huntington. No, it was Smithtown. That the bird was banded the year before at Smith Point. So they moved from the South Shore to the North Shore. And broods are known to move around even throughout the year. Broods have been, I observed, moving 1.5 miles down the beach. So they move around quite a bit, but I don't have exact data on the top of my head.
In terms of compromises between protection and recreation opportunity, does that mean just fencing off certain areas and then the rest of the beach is still available to the public, or what does that compromise?
Yeah, so again, we'd have to sort of start developing that to really know, but it would have to do with less proactive fencing. A lot of sections of beach are proactively fenced off before plovers nest there, and it often closes huge portions of the beach that don't even have nests there. So, a compromise there could be to reduce the amount of proactive fencing necessary, keeping it open, and if the nest does pop up there, nesting it. And again, the habitat conservation plan is a compromise, but it's not, in Massachusetts at least, it's not designed to just open season. It's controlled and highly regulated, and the sites that they decide to loosen restrictions on are carefully selected.
I have 2 questions. Excellent presentation. Are there any gaps in geographically and where the birds are? Like, it all depends on volunteers going out on an ATV or on foot looking.
Yeah, so
Are there gaps in where they're monitored?
Yeah, there are gaps and there are certainly issues with data quality. Again, especially for the productivity number, in order to get an accurate productivity number, you need to be able to identify pairs from one another, because if there are two chicks on a beach and you think there are two pairs, but instead there's actually one pair that re-nested, that'll affect the number. So there are geographic gaps for sure. I know the Fire Island villages are not monitored very thoroughly. And like, I think there was many years where they had 0 pairs, which I don't believe, frankly. I think that they were just missed. So there are definitely geographic gaps, but that's sort of the DEC's job to interact with cooperators and make sure that all the ground is covered.
And there's a lot of beach nourishment and other jetties being built and things like that. And there's also the westward transport of sand, like I live all the way on the other end. We have a huge beach with plenty of room for a lot of nesting birds. I'm just wondering, like, would it be possible to indicate when geographically where certain beach nourishment projects, like, could it actually be a positive impact on?
Yeah, it definitely can. I can't give you like a solid answer.
Really require
yeah,
more.
but I will say, like, for example, Democrat Point, as I mentioned, it wasn't a beach nourishment project, but basically what they did was they flattened out the dunes and just created a flat sand expanse at the end of Fire Island. So if you could sort of replicate that and do a beach nourishment project in a similar fashion, providing habitat, it could definitely, be a good thing.
Any other? We have to be very short questions.
I do actually have, hopefully, oh, gosh, oh, that was what it was. Why did you pick 2001 to start all your trends?
That's just when the data started to be digitalized, I guess. I actually don't know, but working with the DEC, we have all of this data on a hard drive. So that's just the far. I actually initially started, I think 2014, but then I found out that we had it all the way to 2001. So, and other papers have sort of covered, the time before.
The reason I was asking is because sometimes there's like legislative marks that make us choose when we start a trend. Like maybe there was a new conservation effort, and so we start the trend after that. That's why I was asking.
Yeah, I'm not sure. I think it's been pretty consistent since around that time.
Thank you.
Thank you. Hi, I'm Allie. So this is probably gonna be one of the first projects you'll hear about Shinnecock Bay today, but you'll hear many more. So I'm just gonna give you a brief history. Shinnecock Bay is a shallow lagoon in Barville Estuary located on the southeastern end of Long Island, and you can see it on the map there. It's split into the east and west bay sides, and it's typically delineated by the Ponquag Bridge landmark, and you can see it in that picture on the right. The east is generally characterized by good water quality, while the west was notably poor, and that led to the creation of the Shinnecock Bay Restoration Program, or SHRP, in 2012 by researchers at Stony Brook, including Ellen, who created hard clam sanctuaries, oyster reefs, and seeded eelgrass beds in hopes of restoring bay health, which has been successful since. My project involved looking at data from 2 sampling techniques in Shinnecock Bay. The first was trawl surveys, which have been conducted from 2011 to present date. This provides a long-term standardized data set aimed at understanding the fish and invertebrate communities in the bay. And it's also looking at the size, abundance, and distribution of the species caught in the trawl and their potential responses to restoration. The second method was eDNA, or environmental DNA, and this involves DNA collected in simple water samples in the form of slowed cells, scales, and other secretions from marine organisms. And it provides a biodiversity snapshot and allows you to see what might have been in the water at the time of collection. So previously, literature has primarily focused on terrestrial or aquatic environments rather than marine. So it's a novel technique that is becoming more popular in the marine world. And because SHRP also conducts trawl surveys, it provides a unique opportunity to compare a novel technique against a traditional sampling method. So the methods for the trawl surveys, at each site, we go out on the boat and we collect environmental data using a YSI. So we're looking at water temperature, salinity, and salt oxygen. We also record a GPS point at the start and the end of the trawl so we can see exactly where we were in the bay.
The trawl time itself is either 3 or 7 minutes, depending on the location. And after we pull the net in, we start identifying species and recording the morphometrics. And we look at the total length, the fork length, width, and reproductive status as applicable. And we also identify and record the biomass of macroalgae caught in the net. And these are some pictures I took from the trawl surveys this year. The first photo is when they pull the net in and they take all the species out. And that picture actually is from earlier this month when we caught 95 blue crabs in one trawl, which is pretty impressive. And then we record the length either on the table or for crabs, we use calipers. So for eDNA sampling, we collect from 2 different locations, or one we do on the trawling boat at the trawling sites. And for that, we use an extension arm, which you can see in the left picture, usually because the trawling boat is big. And then we also sample at the oyster reefs, and we do that by hand. And then the next 2 days are spent in the lab. So we filter the water on the first day, and we run around 1,000 milliliters through the 0.45 micron filter. And then in the second day in the lab, we extract the DNA off of that filter, and then we prepare the samples to be sent out for DNA sequencing. So the data analysis for my project involved 4 main components. First, I was looking at the life history of 7 species of interest, and I'll talk more about them specifically later, but they're all fish species that were abundant in the trawl surveys, representative of different trophic levels, and important to New York's recreational fisheries. And I also examined the presence and or absence in the eDNA samples versus the trawl data over the selected survey period, which was 2012 to 2022. And then I utilized catch per unit effort, or CPUE data, which is the number of individuals caught for the effort in minutes trawled. So it's how many fish we caught per trawl. And then by year, site, and summer month, and that's July to September. And lastly, I looked at the average total length data for each species by year, site, and month. And also, just as a disclaimer, the biomass and average size of some species could be underestimated due to gear selectivity. So these results were interpreted with that in mind. So the next slides have some basic details about the species that I was looking at, and also some trends that I noticed in my project. So the first is black sea bass. They reach sexual maturity around 23 to 33 centimeters, and the ones caught in the trawl surveys were generally smaller than that on average. And interestingly, they're protogynous hermaphrodites, which means they start their life as females before changing to males at sexual maturity. And this indicates that almost all individuals caught in the trawl surveys are female, and it gives insight into the distribution of females in the bay. And that could be important during periods such as the spawning season, which is from January to July. Next is bluefish. They reach sexual maturity around 38 to 51 centimeters, which indicates that a majority of those caught in the shrimp trawls are juveniles. However, we have caught some large adults in the trawls, and they're usually caught in the east. As you can see by the average size, it's 7 to 68.9 centimeters. And bluefish displayed an interesting trend of decreasing CPUE by month. So the highest was in July and the lowest is in September. And that's pretty interesting because they're a highly migratory species, and this may have implications about their migration patterns out of the bay. The next is a species grouping of scup or porgies and spot, and this is due to some potential misidentification in previous trawl years. So I aggregated the data due to this uncertainty, and you can see them both next to each other in the picture over there. The top is a porgy and the bottom is a spot. So both species reach sexual maturity around 20 centimeters, and it's likely that most individuals in the trawl are juveniles, and they are both influenced by season in some way. Scup are highly influenced by water temperature, and spot migrates seasonally. However, the highest CPUE was actually found in September, so it'd be interesting to look at this in further detail and also with the environmental data that we collect on the boat. Okay. Summer flounder reach sexual maturity around 25.4 centimeters, and that's around the average size in both the east and the west. So unlike the other focus species, summer flounder caught in the trawls are mostly adults. The average CPUE of summer flounder was lowest in September, and that's consistent with their fall migration for the spawning season. And identifying the size frequency and potential timing of migration allows us to look at characteristics of the summer flounder population in the bay. And we can also assume that they're probably one of the higher trophic level species. So the next is tautog or blackfish. They reach sexual maturity around 26 centimeters, and again, it's likely that most of them are juveniles. And interestingly, the average CPUE was the highest by far at a particular site in the east called Cormorant Point. And unlike the other species, there seemed to be a large CPUE difference between the east and the west. The Cormorant Point in the east in general may represent an important habitat for tautog in the bay. The next is weakfish. They reach sexual maturity around 25.5 to 28 centimeters. And again, we mostly catch juveniles in the trawls. They are rapid growers and they typically reach sexual maturity around each one. So this led me to believe that most of the ones that we catch in the trawl are young juveniles. And then weakfish typically migrate and spawn in nearshore habitats. So Shinnecock Bay could be a valuable nursery ground and habitat for juveniles before they transition into adults and migrate offshore. And my last one is winter flounder. They reach sexual maturity around 25 centimeters. And compared to summer flounder, winter flounder are a lot smaller and primarily juveniles in the trawl surveys. And then you can see in the picture, the winter flounder is one on top and the summer flounder is one on bottom. They're both pretty small in that picture. And so they migrate nearshore in winter for spawning and inhabit nearshore estuaries and protected embayments from their larval to juvenile stage. So like weakfish, Shinnecock Bay could represent an important spawning ground and habitat for larvae and juveniles until they mature into adults. And next I'm going to talk about some of my figures. So this figure is showing the eDNA versus the trawl presence. Trawl is in blue. You can see that across species it kind of varies, but eDNA is pretty standard across the board. And most of them were seen in all 3 years, except for scup, which were not found last year, and they were actually found in the trawl. So it's interesting that they were found in the trawls and not in the eDNA. Okay. So this is showing CPUE by site. The east is on the left and the west is on the right. The CPUE is a lot higher in the east, and just for reference, the scale of the west is around 1/3 of that of the east. And at most sites, you can see that the gray color is the dominant color, and that's actually scup in spot. And then this is just a CPUE by month graph. Again, you can see that scup and spot make up the large portion of that. And you can actually start to see that the CPUE increases over the summer season. So the lighter color is July, and then it goes up to September. And then these are the other 6 species without the scup and the spot. And you can see that some of the species, again, are displaying trends. And in my project, I looked at these in like a lot finer detail on an individual level, but this is just an overview. So I chose this capstone project because I was really interested in the use of eDNA as a non-invasive novel technique. I also think that evaluating the current conditions and characteristics of recreationally fished species is important, especially because restoration typically focuses on lower trophic levels first, like forage fish. And however, these species directly impact New York recreational fisheries and fisheries management, which is why I wanted to focus my capstone on them. And this type of information can be expanded upon and used to inform future ecosystem modeling work in Shinnecock Bay, and that can in turn contribute to ecosystem-based fisheries management techniques. And that's it. Is there any question?
Yeah, I was wondering if you knew like how much eDNA was being used currently in like management of these species at any of these agencies.
Do you mean like data that's informing management practices? I'm honestly not sure. The study that I read was talking about how marine made up like half of the amount of like aquatic studies that were being done using eDNA, but I'm not sure exactly how many practices are being informed by eDNA at this time.
Other questions? Okay, great job. I also want to acknowledge Gordon Taylor, who has made his lab available to us for doing the DNA work. And thank you very much, Gordon. Okay, next. So, you know, for those of you who may have left the room for a second, we're moving a little bit faster, but we skipped over one of the presentations. So the next speaker will be Corinn Kraemer. And her faculty mentor is Chen, and she has worked with NOAA for her internship.
Use that mic or you want?
Yes. So, the title of my internship is Water Quality and Submerged Aquatic Vegetation Planting in the Hudson River near Stockport, New York. So, just some background on the Hudson River. I was not too familiar with the Hudson River when I first started. Now I am a whole lot more familiar. In total length, it's 507 kilometers long, with the first 240 kilometers of that making up the estuary itself. As Casey mentioned, it is tidally influenced, and they have seen tides of around 1 to 1.5 meters in height. My supervisor Sarah was saying in some areas they get up to 6 feet tides. So like us, they have tide changes every 6 hours. Salinity influence is a little different for them. Although they are fresh water, there is some salt water influence up the Hudson, so it'll extend 40 kilometers to the north from the mouth during significant discharge. So if there's a lot of rain that year and the tributaries are contributing a lot, the salt line is much lower. But okay, we got some covering here. If there's not a lot of rain that year, it'll extend 120 kilometers north, which is around the Poughkeepsie area. And the shoreline ranges along the Hudson from riprap to revetments, which is piles of boulders and rocks that are shore stabilization. A lot of this is seen around the rail lines, to sandy and rocky beaches. So this is the main submerged aquatic vegetation of the Hudson, or SAV. It is Vallisseria americana. It's also known by common names such as water celery, wild celery, or tape grass, depending on which paper and source you look at. It is capable of reproduction via pollination and seed-bearing fruits, but is also capable of asexual reproduction via stolons. So if you've ever heard of Zoster marina or eelgrass, they kind of can bud the same exact way. They have a rhizome that branches off. It's a similar concept. These plants actually can survive the winter. So they produce a tuber at the end of their roots. And while the plant on top may die off, that tuber to start the plant next year stays in the ground over winter. And beds are known to have 4 to 5 times higher density of macroinvertebrates, so it's a very important critical habitat for certain species. And unfortunately, true color photos and ArcGIS analysis have revealed a 64% decrease in the beds from 2000 to 2007 to 2014. And they link this a lot to the tropical storms and remnants of hurricanes that we had from 2011 to 2012. So Irene, Sandy, I forget the third one's name, to be honest with you, but so the Hudson River National Instrument Research is located at Norrie Point. Environmental Center is in Staatsburg. And my internship was with them, with my supervisor, Sarah Fernault, who works with subaquatic vegetation and also marshes as well, the freshwater marshes that they have along the Hudson. So they have a main objective to restore habitat along the Hudson. and also to help progress conservation targets as they have prioritized in their Hudson River agenda. So for me, I also wanted to do something a little extra, just more than contributing as an extra set of hands. I wanted to examine water quality parameters. So large SAV, so there was a study done by Caracco and Cole in 2002 that large SAV beds actually had pretty good dissolved oxygen levels. They never reached below 5 milligrams per liter most of the time. But another species that they're having issues with the Hudson, Traspinatus, which is water chestnut, it's not necessarily very good for their dissolved oxygen levels. So if they can restore the beds, it may help to improve local water quality in the area. So I wanted to test to see if we could see improvement in the caused by the plants that were planted in Stockport. So that my hypothesis for this project was planted Vallisneria americana would significantly improve the water quality at Stockport, and the null would be that we wouldn't see any significant improvement. And I only chose 2 water quality parameters because they have, and I'll show you in a second, they have these data loggers, and they do everything from temperature to pH, salinity. It was just a massive amount of data to do everything. So I picked 2 that I thought that both me and my supervisor thought we would see the most improvement in, and we tested those. So I'm going to introduce you to one of our study sites. This is Tivoli Bays. It is primarily a freshwater marsh, and it is one of the sites of their longest term, long-term monitoring sites. It houses a YSI data logger. If you hear me refer to it as a sonde, that's what the more technical term for it. It's also home to Kruger Island South, which, if you can see down in the left-hand corner, those blue lines are 21 long-term SAV monitoring plots, specifically in a submerged Vallisneria bed that is natural. There was no planting done here. So it was selected due to the proximity to its sonde and the natural bed itself, and the transects. This is Stockport Flats. This is where the plants were planted. It is a variety of habitats. It's got mud flats. It's got a little bit of a freshwater marsh. So it really does range in the variety of habitats that it houses. Stockport Creek is one of the biggest tributaries to the Hudson. And the area is used by fish as spawning grounds. It's used as birds for nesting and hunting grounds. So it is important to not just aquatic organisms, but also more terrestrial based and organisms. Most of the Vallisneria in the past have been found in the subtidal shallows that feature in Stockport. So unfortunately, I did not get to help out with this part of the project. The actual planting occurred before I came to Norrie Point because they had to be put in May, and I arrived in June. So this site was chosen pre my arrival there to receive the water celery. This year, they only put about 10% of what they want for their 3-year plan, which totaled out to be about 20 plants. So each transect line received, the 2 circle transect lines received 10 plants within that little red circle that you see. And this was done by putting the plants, the plants came from the Appalachian Lab at the University of Maryland Center for Environmental Science. They were then put into these biodegradable bags, and they were staked to the ground in the area using a snow pole. As I said, this was along 2 transect lines. The one furthest bottom left that circled received 10 the plants there received 10 exclosures. And this was to prevent herbivory. Wow, I can't speak today. And that was just to test to see if it would have any impact on the plants growing. It was kind of just like something on the side that we threw in. This was accomplished by taking sections of snow fencing and 2 other poles. We kind of tented we pulled the cone, they were cut into cylinders, and we put the cylinder around the poles. We put them into the ground a little bit, and then we zip tied the top off to kind of make this cone shape. So my analysis involved surveying both sites for coverage in August typically, this was done August 1st, but typically this gets done a little later in the year, which means that I may not have perfectly accurate coverage for Tivoli and Stockport, but because of the time restraints of my internship, it was done in August. We also I measured water quality using the pre-existing sondes that they have at the site. So Kruger Island South used Tivoli Bay's sonde, and Stockport used Ferry Landing. And right on the right, on your right, you can see, this is I'm not sure if this is the current version they use, but this is a display sonde that they have of what they actually use. And then on the left is one of the structures that this sonde is put in. This is outside Norrie Point. I'm not sure that the one in Ferry Landing and Ferry Landing and Tivoli South are installed the same way, but because there's a dock right there, this one's extra supported, but it still needs to be protected. My data came from the National Estuarine Research Reserve Centralized Data Management, which is available to the public. You can click on this link and you can access live feed from these sondes. Literally, you can pop on right now and say, oh, what's the pH in Tivoli? And it'll tell you the pH in Tivoli. Or you can graph what was going on the whole entire week. So for my actual analysis, we used Microsoft Excel. I calculated daily averages and standard deviations to get an idea of what the average, let's say, dissolved oxygen for Tivoli South was on August 1st. I used graphs to then graph them for the time span that I selected, and I used t-test to sample assuming equal variance to evaluate the significance between. So these first two slides are the pre-planting water quality, so dissolved oxygen and turbidity. So this one popped up a non-significant difference between Tivoli and Ferry Landing when it comes to turbidity. So there is no significant difference. This one is a little harder to tell where everything is, but this is dissolved oxygen for both sites. This one did however, come up with a significant difference between Tivoli and Ferry Landing, with Ferry Landing having higher dissolved oxygen than Tivoli South. This is after the plants. Something really, I'm gonna mention this now, something really strange happened with the turbidity for Tivoli South, and there is a possible explanation for this that I'll get into later, but this one did pop up another significant, this after planting Breda popped up a significant difference between Tivoli and Stockport, Ferry Landing, Ferry Landing, with Tivoli having significantly higher turbidity. And this is dissolved oxygen. So this one was extremely significant difference with Ferry Landing having higher dissolved oxygen than Tivoli South. So as far as the plants go, which that on the right is a picture of a bed in Tivoli Bet and Tivoli actually in Kruger South during one of the transect surveys. Stockport unfortunately didn't take so well. It was almost 0% coverage. Yeah, I'm gonna Kruger Island had a 20 to 50%, as I stated, with the pre-planting and after planting. I'm just gonna say this really quickly. It's very hard to link these improvements to the plants, because they just didn't survive very well. But also, the one that stands out to me is the distance from the sonde for Ferry Landing is 3 miles. So it's very likely that that sonde is picking up a signature from a local bed rather than 20 small plants 3 miles away. There are some improvements that they can make. It is useful for habitat restoration in the sense that you can tell, it tells what doesn't work. And water quality and valve examination is useful to further the knowledge about what parameters they do improve. These are my acknowledgements. I would like to thank these people. Without them, it would not be possible. And questions?
Maybe one question?
Nobody likes being out of time.
There you go.
What would you say, you think the most significant reason is the vegetation at a different time established?
So I didn't put this up there, but when we were surveying Tivoli South, I realized something very quickly about Tivoli. We survey typically during low tide because it's easier for us to get in and out during low tide. Tivoli South during low tide is about 3.5 feet in depth. The transects in Stockport, it depends on where you're sitting on the transect. It can range from you're high and dry to 25 to like 15 feet deep. So the transect spot for the plants was only about 6 inches. So it's probably a depth concern. And that's one of the improvements that they can make to it is to push the plants deeper to see if that's what's affecting them.
Okay, well,
Hold it up a little.
okay.
Maybe,
Or
or just
to
pull
this
it up.
extent.
Nope,
No?
nope.
Okay.
Sorry.
Sorry, guys. Well, so thank you for that introduction, Ellen. So again, my name is Hayley, and I'll be presenting my capstone titled Comparison of Antarctic Cetacean and Pinniped Sightings from Tourism Vessels and Scientific Literature. And again, I did want to thank Gina and Heather Lynch for being my advisors. So in this presentation, I'll be discussing the motivation for undertaking this project, my objectives that I was achieving, the approach I used, the results and products I created for IATO, and the broader significance and impact of this project. And so historically, marine mammals populations across continental and maritime Antarctica were driven to near extinction by overharvesting. And so, for example, Antarctic blue whales that had once numbered 239,000 before commercial whaling in the 1904-05 austral summer season were reduced to just about 360 individuals by the 1972-73 season. And so similarly, fin whales were decimated to 2% of their original numbers, with fewer than 6,000 individuals remaining in the Scotia Sea and the Antarctic Peninsula marine ecosystem. And so following all of this extensive commercial hunting, the protocol on environmental protection of the Antarctic Treaty was put into place. And it was created to protect the continent as a natural reserve dedicated to peace and science. And this legislation came into effect in 1998. And so Annex 2 specifically focuses on protecting Antarctic plants and animals from take or harm, and other than for the purposes of scientific research or official collections. And so with these no-take regulations in place, tourism has become one of the primary non-consumptive activities that occurs in Antarctica, specifically around the Western Antarctic Peninsula, or WAP. And so what does this have to do with IATO? So the Antarctic tourism industry has seen a 50% increase in visitors from the 2014-15 season to the 2018-19 season, and this increase is likely to continue. So to put this into perspective, this year, during the 2022-23 field season, IATO hosted 105,311 visitors with 32,730 cruise-only visitors, 71,238 landed visitors, and 1,343 deep field visitors. And so given that whale populations are still recovering from commercial hunting, there are high population densities of whales around the WAP, and there is a high risk of vessel strike where there are whale watching industries involving small vessels. IATO is concerned with reducing this risk in the areas where they operate. And so to reduce the risk of fatal whale strikes in Antarctica, IATO members voted in 2019 to create 2 slowdown areas on the WAP based on high numbers of sightings reported in Happy Whale, which is a citizen science photography archive for marine mammal sightings. And so these 2 slowdowns are shown in that top figure on the right. And the image below shows the density of sightings from Happy Whale. And I kind of estimated the boxes just to show like how many sightings were occurring in those areas. And so both zones that passed encouraged traveling at a minimum or maximum speed of 10 knots in those areas from January 1st to May 1st. And so these were preliminary measures in 2019, and IATO recognized that they would need to be amended in the future to protect whales for a longer part of the tour season or to protect like other dense areas. And so to determine if these areas needed more protection for a longer part of the tour season, IATO initiated their first season of the Voluntary Cetacean and Pinniped Sightings Program, which is VCAPS. And each week the operators were asked to submit all of their marine mammal sightings, which provided evidence for proposing new slowdowns this year at the 2023 annual meeting. And so as their VCAPS intern, the objectives of my internship were to provide IATO members with weekly updates regarding everybody's collective marine mammal sightings across the WAP, then to use GIS to map marine mammal sightings in preparation for the meeting, to compare these sightings to what was known in the scientific literature, and then to make recommendations to IATO regarding the future of the VCAPS program and what information from the literature should be shared with operators. For example, if there were sightings in different places that they didn't go to, or other differences between what their data said and what is there. And so from November 2022 to March 2023, operators recorded their marine mammal sightings in spreadsheets with the time, the name of the vessel that the animal was spotted from, the name of the observer, platform, species, whether it was cetaceans or pinnipeds, and all that information, and of course their coordinates. And so to incentivize operators to participate in this program, I was tasked with preparing weekly maps to provide to these operators. And so I made 2 different PowerPoints every week, one with cetacean sightings and one with pinniped sightings. And each PowerPoint had 5 cropped images from this map on the left of the different regions of Antarctica. And then I converted the coordinates into decimal degrees and used Google Maps to estimate where they would be on those images. And so the top image on the right shows the weekly maps of the Gerlache Strait that I made from February 6th to March 19th. And as expected, each week there were a lot of humpback whale sightings, especially sightings that had more than 3 individuals. And so the image on the bottom is just an example of a table that I created to summarize those sightings. And then I also noticed from those weekly maps that there was a high density of cetacean sightings around the South Shetlands and Elephant Island group. And so to prepare for the annual meeting, I was tasked to map all of these sightings in the area from November to March and plot hypothetical slowdown zones. And so the yellow shaded area on the left represents one large slowdown zone, and the 2 red areas on the right are smaller ones. And so I used proportional symbols to emphasize that there were several large aggregations of fin whales occurring around Elephant Island, making this area a strong candidate for a new slowdown zone. And so both of these maps were considered by the IATO Marine Committee, but they were not practical for operators because they covered an area that was a little bit too broad. And so in addition to the combined with the data from ORCA, Ocean Research and Conservation Association, and the University of Tasmania, members modified that secondary proposal into a final slowdown zone that's shown on the right. And so this new proposal covers 12 nautical miles surrounding the South Shetlands and the Elephant Island group, and addresses safety concerns with respect to the Drake Passage, while also considering the bathymetry around the islands, which was important for foraging fin whales that were there. And so in addition to these weekly maps and the annual meeting maps, I also created additional maps of total sightings of cetaceans and pinnipeds through the entire season. And so the map on the left shows all of those cetacean sightings with approximately 1,300 cetaceans, most of which were humpback whales, and only 1 blue whale the entire season. And the map on the right shows all of the pinniped sightings, which I plotted nearly 700 with mostly Weddell seals. And so based on my literature review and combination of reviewing those maps, my first recommendation for IATO was to continue the VCAPS program into the future, because there was a pretty big lag time in the literature between when their data on sightings was collected and when they were published. And so, for example, one 2022 study used data on Antarctic fur seals on King George Island that was collected from 2011 to 2018. And then even then, the study only got published in 2022. And so there was a really big time between when the last data was collected and when it was published. And so additionally, IATO uses adaptive management to regulate the tourism activities of member vessels. And adaptive management is the structured process of learning by doing and adapting based on what is learned. And so while a 2021 paper suggested that this process should depend on the scientific community for assessment, design, monitoring, evaluation, and adjustment of these management measures, this can be made unnecessary by collecting their own data. And so many studies in Antarctica use tour vessels as platforms anyway. And so using their own source data would make the organization more quickly and independently change their management strategy to more effectively manage tourism. And so my second recommendation for IATO is to make participation in the BCAPS program mandatory and offer incentives to increase operator participation. And so this season, an average of 13% of operators participated in the BCAPS program, with a low of about 9% and a high of about 20% of operators. And some ways to incentivize these operators to participate could be to waive their membership fees or reduce the fee by taking out the component of like $3.50 per person per passenger when the vessels are south of 60 degrees. Or they could give early access to Ship Scheduler, which allows the vessels to decide which parts of Antarctica they're going to on given days during the tour season. And so these incentives could be given to operators that participate the most frequently in terms of how many trips that they went on, so that like we don't encourage people to make false reportings. So if an operator submitted like 10 of 15 of their cruises, where another only submitted for 3 out of the 8, they would, the one with 10 out of the 15 would have a better chance at receiving that incentive. And so absences of species in areas where they're expected could also be tracked to reduce the chance of operators changing their route to encounter more mammal dense areas. And my third recommendation for IATO was to share what I learned in my literature review about crab eater seal distribution. So operators noticed that this season there were a lot fewer crab eater seals on their routes than in seasons past, and they notified the IATO Secretariat. And so I found this study by Huxat et al. In 2020, who modeled the changes in predicted crab eater seal habitat importance under future environmental conditions around the WAP. And so they found that there was going to be a significant decrease in habitat importance around the South Shetlands during the whole tour season from November to March, and then in the Bransfield Strait north of Brabant and Anvers Islands in October and November. And so they also expected areas beyond the Antarctic circumpolar current and more south along the peninsula, which are less traveled by operators, to have increases in the presence of the crab eater seals. And so this is just something that they might not see in the future. And so this project has broader significance because tourism is an exponentially increasing industry with no signs of like slowing down. And so cruise tourism is the largest growing sector of this industry, and increasing cruise tourism increases the risk of vessel strikes for cetaceans. And strikes are one of the greatest anthropogenic threats to whales worldwide. And for Antarctica, it's really important to mitigate these impacts because marine mammals are again still recovering from historical hunting. And so this project contributed to marine conservation and policy by helping to create the 2 new slowdown zones seen here at the IATO annual meeting. And we learned about how data collection by operators enhances their ability to use adaptive management to conserve marine mammals, as changes can be seen more quickly than they can be in the scientific literature. And so with a quicker turnover rate in data, they can expand policies that achieve their goals or discontinue programs that do not show a positive impact on Antarctica or its wildlife. Here's all my references. And
project is all of the tourism activity confined to the peninsula region?
No, most of it is on the peninsula, but there are some operators that operate in like East Antarctica and go to like the sub-Antarctic islands, including like South Georgia and the Falklands.
Section 9 as well.
Yes. Yeah.
Okay, so I'm Heather, and my project is monitoring a fecal fiasco, sampling to Save the Sound. I worked with Save the Sound. So first I'll go into who they are. They're a non-for-profit organization with the goal to fight climate change, save endangered lands, and protect the sound and its rivers. They have several strategies for achieving their goals. They include climate resilience plans, protecting and preserving the natural environment, ecological restoration efforts, and working towards healthy waters. Healthy Waters is where I was involved with the bacteria monitoring program, as well as there's the Unified Water Study, which I'll talk about a little bit. Some of their other campaigns right now, they're aiming to preserve Plum Island as one of their natural environment protection goals right now. They are doing ecological restoration efforts, including dam restoration, establishing fish ladders in local rivers.
This is my advisor and the environmental analyst, Amira Khan, in her mosquito suit. A big part of what I do is going around and sampling on Monday, Tuesdays, and Wednesdays. And a lot of the sites we go to are very, very bug infested, and you have to fight a lot through the brush, and there's a lot of animals and insects that we deal with. So mainly what I'm involved with is the bacteria monitoring program. This is a 12-week long summer program going from June to August. It was kickstarted in 2013, and the aim is to quantify the concentration of fecal indicator bacteria in the sound, in rivers, tributaries, and along the embayments. We test for 2 types of bacteria, Enterococcus and E. coli. Enterococcus we look for in brackish waters. It fails anything above 104 mpn, the most probable number of colony forming units. And for E. coli, which we're looking for in freshwater sites, is 235 mpn. We test for both in a few locations because say one day we share this information with the DEC or the EPA, we want to have both on record because depending on the salinity, we might call something freshwater that they really think is brackish or vice versa. So for a couple of the sites, we test for both. The bacteria indicate the presence of untreated sewage in waterways, a big issue in the Western Long Island Sound, especially near the city, where combined sewer overflows occur during storm surges, and that just washes in treated and untreated wastewater into the waterways. So that's what we're mainly looking for. In some settings, more agricultural settings, that runoff could be from fertilizers or livestock, but since we're in a more urban area over there, it indicates the presence of sewage in the water. And the collected sample results are used to produce maps weekly, and then at the end, more cohesive data sets. It is still the monitoring season right now, so we don't have our final data sets, but we do have our weekly map updates. And sample collection relies on a joint effort between volunteers and Save the Sound staff. So every week I go out as well as the volunteers and our other water quality staff. Here's a couple pictures of sample collection. That's me. I was on their Instagram page for National Water Quality Month. So as you see, you get your little grab pole and you do samples and you reach into the water. I'm in a river on a bridge at this site, or you can see in the bottom picture off of a dock. Some are private properties that we have permission to go to. Some are public beaches. Some are just lakes, not lakes, rivers and tributary sites. So the first step is to collect the samples. Everyone is given maybe 2 to 4 sample sites, and we sample different locations, and I'll have to show you the map and show you sites that we do on certain days. After that, we do sample preparation, which I'm also a part of. That includes doing sample dilutions. We take 10 milliliters of our samples into 90 milliliters of water, and we mix it with our reagent. Depending what we're looking for, we use different reagents. We use Enteralert for Enterococcus or Colilert for E. coli. And so each one gets mixed with that reagent and then put into these trays where we're labeled with the sample times and the dates and what our target bacteria is. It is then incubated, depending on what that bacteria is, Enterococcus for at least 24 hours to 28. E. coli is a little shorter, 18 to 22 hours is what we're looking for, is when we look at it afterwards. And then we analyze the samples. I, myself, or the other water quality intern will do that. Can't really see in this, but this is the chart we use based off of our IDEX trays. So we're looking for which cells are fluorescing, and then we plot these small wells that are fluorescing against the large ones, and we get a number, dilution factor of 10, multiply those numbers by 10, and we get the MPN, the most probable number of bacteria in that 100 milliliter sample, which is our original sample size. And then in this image in the corner, you can see my little data sheet, and I use that data and I compile it into a master data sheet, and then weekly, we use that master data to create the maps. Here are more samples. Here's me wading through a site that has a 100% fail rate for Enterococcus, but there's 1 week left of testing, so who knows, maybe this week is the week it passes. This site is the side of the Cross Island Parkway in Queens. So on Mondays from 10 to noon, if you're ever driving there, you could see me crawling down those slick rocks at low tide with, I've had a lot of experiences at that site specifically. That's where that is, right before Bayside Marina. Here are 2 examples of maps. One's a wet weather week and one's dry weather, and that means if there's more than a ½ inch of rainfall between our testing day or 3 days prior, it is a wet weather week. And this can really show the impacts of wet versus dry weather on our sample collection. So one of my other responsibilities is collecting precipitation data for the given regions for the days that we test. So Mondays, we go to 2 places. We do our clean sites and we do our Greenwich sites. I, as the only Long Island resident that works here, meet our 1 volunteer here and we do all of these sites in Queens, and then everyone else and our volunteers go to Greenwich on that day. Tuesdays, we go through New Rochelle, and Wednesdays we go through the Mamaroneck. Our office is based in Larchmont, so it's here's one covered by all these dots. And that is where our lab is. So wet weather week, you could see a lot more fail sites. This one is actually, this is our most recent week that I had access to, the 14th to the 17th. This is actually the most passed sites in a wet weather week that has like ever happened for Queens. We have 4 passing sites during wet weather week. Usually before this, the most you would get is 2 passing sites in Queens on a wet weather week. And then as you can see, a lot of fails. Orange is a fail, red is 10 times the failing standard set by the EPA. So a lot of those. And you could see a dry week, you get way more passes, way less 10x fails. Seems this one only has one in Queens on this day. Oh, there's one hidden in there too. Another thing I got to be a part of is the Unified Water Study, and that was established in 2017 in order to create a common protocol and a comparable data set for water quality parameters throughout the Long Island Sound. This focuses a little more, rather than just our embayments, a little more open water. We sail around, we have a little ship, and we get to take readings out in slightly more open water rather than just being restricted to the coastline. So local groups are trained and given materials and are included on our quality assurance plan, and we measure for different parameters. I got to go out one day, my thing got cut off here, but it reflects the health of the sound as well as human impacts on the sound. We go out of several different ports. I got to help leaving from City Island. We get to go out at sunrise because a lot of things are photosensitive, so we try to go out within 2 hours of the sun rising. Here are the pictures I took that day. Here's our portable sampler. Here's a really nice sunrise picture I took at the marina because I didn't wake up at 4 for nothing. And this is our seasonal tech, David, using YSI probe to log some of the parameters, and I got to help. I was tracking coordinates and writing down our readings at surface level and at depth. And the idea of this is because a lot of groups previously are using different methods, doing different, like testing at different times. So we created a common schedule and protocol so that we can create a comparable data set for the health of the Long Island Sound. Another thing that I was in charge of, or at least helped in overseeing, was the volunteer activity. Volunteers are a major role in our bacteria monitoring season. We have, I think, 62 sites, and it would be impossible for us to do this without the volunteers. So we have their availabilities and schedules, and I would help us make those schedules. So we have these little Excel sheets, and then what days they're free, maybe what times they're free in those days, and then based off of that and the proximity of sites to one another, we create these chain of custody records, and on each record, it indicates where they'll be going. And there are site sheets associated with each one, so they know where they're going, if there's any special parking they need to do, or if they need to walk or hike anywhere. And then they fill in their coordinates that they sampled at, their times, they can do any observations that they were seeing. And then they're signed out by me, in by the volunteer, and then signed in at some other point back to the lab. This one was signed in twice because we had to go out and do an extra sample on this one. But with that, we can keep better track of who took what samples in case there's any problems or we need to refer back to them. And with that, I don't know, okay. And with that, we use the data collected from these to make those weekly maps I showed you. And the bacteria monitoring season is ongoing. So this coming week will be the last week. I will continue to work with Save the Sound into September. So this will be our last actual week of sampling. And then past that point, we will produce geomeans, we will produce statistical and statistic records to compare our data this year to historical data from previous years. This is from last year, so this is just an example of comparisons we can make. Like we can see that last year our river site health is declining, and this can help us focus efforts on restoration or management practices for certain sites like our rivers. That site that I was wading in is a tributary feeding in to a larger river feeding into the sound. So we're seeing a lot of bacteria increasing in concentration over there. So maybe that's something to look for. We can see if certain areas are really, really bad, if they're not normally bad. I was sent to Cold Spring Harbor one day because we had a report, kind of a watchdog program we have, where someone saw something that looked like maybe a slick, maybe some kind of spill or something. So I went out to Cold Spring Harbor that day and did some samples there at our exploratory sites, which we might not normally do otherwise. Overall, we could see on the right side of the screen, our failure rates are increasing, at least last year. So when we get this year's data, we can compare that and see if they're still increasing that fail rate, or maybe if we're bouncing back a little bit and measure the health of the sound a little better. So ask me in 2 weeks if we are still on the decline. But right now, that's as much as I have. And I would like to thank my advisor, Mary, and as well as everyone at Save the Sound, Amira, and the rest of the water quality team.
Okay, looks like it's time for a couple questions.
Great job. I don't think I caught how quickly the samples are analyzed in the lab, and do they inform beach closures? Like, are they fast enough so you can say that it's not safe to swim somewhere?
Well, those samples from the week are not reported till the end of the week, so it is public access as soon as they're up, which can influence beach closures. But samples are collected and they have about an 8-hour max, we call it 6-hour holding period. They are processed and they need like a day to incubate, give or take. So our Monday samples are then analyzed on Tuesday, Tuesday samples Wednesday, Wednesday on Thursday. So then by Thursday, we have our total data set for the week.
Do your surveys overlap with New York State Department of Environmental Conservation or New York City DEP?
So I don't know, they want to work with the DEC and like send our samples to the EPA. So right now, I think we do use similar sites, but I don't know. I think there's only minimal overlap in where we're testing and further testing.
It's the DEC that determines that.
I think we do send our data, and they want to send it to EPA beyond, but not yet.
I have a quick question. How do you keep yourself from getting sick?
Didn't you see my waders? Those are top of the line. I don't know, very important. We express the need to wear your proper gloves as to not contaminate samples and to kind of keep yourself out of the water as much as possible. I've definitely dunked into some pretty disgusting areas. The Cross Island Bay sites are pretty.
Have you been healthy this whole time?
I have been. I think it makes me stronger. My immunity has shot up.
That's good. All right, well, thank you very much, Heather.
So hi, yeah, my name's Paris Bard, and I'm gonna go over my internship with Cornell Cooperative Extension, where we did spawning and juvenile stage substrate experiments with rib mussels. Yeah, so just a quick overview. I'm just going to talk about why I did my internship with CCE and rib mussels, a little bit of background on the mussels itself, and then go into the research I did, which was split up into 3 phases. So I did some spawning experiments with the rib mussels. I did a larval stage settlement with 3 different substrates that I tested, as well as with juveniles later in the period, and you'll see why. And then also the significance of this all. So yeah, really quickly, just what the rib mussels are. So they're an intertidal benthic species. It's native. You can find them in New York waters along the coast and along the coastal United States. It has a wide-ranging filtration ability, which is really important because not only does it take out nutrients such as phosphorus and nitrogen that can cause such harmful algae blooms, but it actually filters out bacteria as well. So bacteria that might be in fecal contamination, it can filter that as well. It is found in salt marshes. It forms a relationship with Spartina, so the common salt marsh grass in the area, and it spawns in the summer. But a key aspect of it is it has these byssal threads that it allows it to attach to substrates and to each other, and anchors down in environments that are, that has a lot of impact from waves. So my own motivation, this species has a lot of potential in restoring ecosystems. So we used to have a lot more salt marshes and mud mussel beds than we do now because of either dredging or habitat loss, but due to building up shorelines. And it has a lot of co-benefits along with it. I discussed how it takes out nutrients, so it helps preserve water quality. But it also is important key habitat for species like spawning and small fish and crustaceans, as well as building up the shoreline against storm surges that we're going to see more common, more extreme due to climate change. And yet little research has been done compared to, say, oysters and clams in the hatchery rearing juvenile stages. So as far as the spawning experiments go, the reason I wanted to test this is that there's two main methods typically of spawning bivalves. There's thermal cycling, which is kind of it takes like a couple hours, 4 or 5 hours sometimes to do. It's very successful on clams and oysters historically. And then a bin silo method, which usually takes a matter of days to complete. And that has been successful on ribbed mussels, but it would be better in a hatchery setting. It's more beneficial in a lot of ways to do thermal cycling. You can isolate the spawning individuals, and that isolates the gametes, so the eggs and the sperm, and that prevents over-fertilization, which can damage the larvae, as well as just makes it quicker and more efficient. So I ran 2 thermal cycling trials and 2 bin silo trials, which I will talk about. So the thermal cycle, you run cold water on it initially, let's see. Right here, they're all spread apart. Obviously, it runs cold water. It's just a thin layer. And then you heat that up really quickly within a matter of half an hour. And if you don't see any spawning individuals, which kind of like a white cloud that they puff out, you just reset that. You drain the water, do that again and again. We tried it 3 times, or 3 different temperature intervals. It usually goes from 15 to like 30. And that was... that did not produce a spawn, so we tried it again, and this time the temperature range was more extreme. So that was from 10 to, and then we went up to like 36. And we kept the byssal threads attached to each other. We didn't cut them. So just to see if that affected how they responded to the temperature changes. Still, there was no spawn successful. Going to the bin silos, that was a successful spawn. We had a total of 4 spawns over 2 attempts. And basically you fill this bin up with hot water, usually around 30 degrees, and you put the mussels in it in this smaller silo in the middle, this silo. And then you kind of have the water circulate, and it cools naturally over the night, and you come back the next day, usually typically within like 20 to 24 hours, and then you'll see if you'll have a spawn. You drain it down into a 28 mesh sieve. And yeah, that's one of the more successful. There's a lot of D-stage larvae, so that's the first stage of larvae. It looks kind of like a kidney bean, and that's after 24 hours, as well as possibly some more eggs that haven't fertilized. So I just wanted to really quickly go over the differences visually. So this is our thermal cycle. You can see this is a matter of 5 hours, and there's just these really quick increases in temperature, really quick decrease, and then it goes up again over a span of like half an hour. And so that's what you're looking at with the thermal cycle. It just does not really work with ribbed mussels at the moment, at least the time frames that we did it. But if you're looking at the bin silo, this is over a span of 12 hours that it's decreasing, 24 hours, and it's over multiple days. And it's a much more gradual process. So this is just something that the rib mussel as a species compared to other bivalves, it works well with. Yeah, so just to go over the larvae that we collected, here are some of the larvae you can see under a microscope. The first attempt, 50,000 about, had a regular growth, so I decided to scratch that and just go with some more trials, and it ended up having a really successful third attempt, where we gained the bulk of it. And I was thinking of stopping there, but we decided to go for a fourth attempt, just to see if there were mussels that didn't spawn. Usually they all spawn together because the cue is that there's like the gametes in the water, helps with that cue. So once one spawns, usually the others will start spawning again. And there's actually a significant amount of larvae collected after that large spawn, which is important for hatchery workers to know. If you're doing a spawn on rib mussels, you know, and you have this large spawn, maybe you want to go and do it again, and you're missing a lot of the gametes that you could possibly get out of that brood stock. All right, so just I also did a lot of work in general in the hatchery. You know, I had to keep the larvae alive. I also helped with larvae from clam work and oyster work that Black Spot Marine Lab was doing and Cornell was doing. So over there, that's the algae that we feed to the bivalves, and so we had to maintain algae production. That's something I learned. This is the big conical that all the algae lived in for some time, and this is a rib mussel larvae that just hit its eye stage. So that's about 21 days after it first spawned. And that signifies that it's going to start wanting to settle on substrate. So that's when we get to the substrate part of my experiments. Now, the reason I wanted to do this was that I have a big interest in doing restoration with these mussels. I think they have a lot of potential. And so we wanted to find a way to get mussels from a larvae or from a hatchery environment out into the wild in a way that's like cheap, efficient, quick, and with that, the mussels themselves would most likely attach and work with. So I tried out this, it's called bees, and it's a biodegradable material made out of potato starch, and it kind of layers on. There's a lot of structure to it. So I wanted to see if the larvae would attach there. We also use shells from dead rib mussels, which is a common substrate for other bivalves such as clams and oysters, and sand, which is for this experiment, the control. It's just something that they find naturally in their environment. And so all these bins, they had them at the bottom. This is the bottom of the bin, and they're all identical. And it was going to be 5 weeks that we were going to have that going, except there was an unexpected mortality during that time. So this is a zooplankton, not really sure what species, but it ended up infiltrating the conical they were living in maybe like a week after the spawn. And they were doing fine, so we just kept it going. But it was they eventually, when they were in the bins, they ended up likely outcompeting the rib mussels for food. So here's us looking for the mussel, and we found this is potentially a rib mussel, but it is much smaller than what we would expect to see at that time. It's supposed to be, they're supposed to basically, after like 3 or 4 weeks, you'd see them with a naked eye. We only found 3 of them, so the whole thing was scrapped. And I decided to do this experiment with substrates with some mussels that were already doing very well and were about 5 months old at that point. So I took the same experimental idea with the same substrate. Well, these two are the same substrates, so the bees and the dead shells, but we use conspecific live mussels as the control, something also that's naturally found in the environment. And a lot of the research that I saw has them as a preferred environment for mussel settlement as well. So that was our control, and we had 3 of these bins where we actually had natural seawater come in with the natural plankton of the bay, Huntington Bay, and so we didn't have to worry about feeding. So that was nice. And just let them see what they did. So we have 50 in each. And there's also other, so that's just like aspects of the tank, which I'll get to. Yeah, so looking at, we did 4 counts after it's about a week, so August 3rd to August 13th, about then. And there was shown to be a preference with the live conspecific for the juvenile mussels, as well as a preference for the other aspects of the tank, which I will get into. And these were actually not at all. There was a lack of preference there compared to the other substrates, which is likely due to just like less structure and like aspects where they could hide in it, which I can also talk about. So there was very little movement, but there was slightly some movement. So these were the days counted. August 4th, and so they pretty much settle where they want to for the initial first couple of days. But there was a little movement you can see into live conspecific, and as well as into other, and the dead shell really they kind of push back from that. So that's a little interesting. They have a preference there, and they'll move around. Some of them will. And so looking at the other aspects of the tank, there's a on the bottom of the bin is where the water comes in, and so there's a layer of mud that usually accumulates. So I would consider that to be like the bin mesh, but also mud. And then under this drain is the outpouring drain at the top of the bin. They liked being underneath there, or some of them did. There was compared to the bin side, which was just the plain plastic. And an idea of why there was a preference shown for the mesh mud is that it kind of gives an example of a natural environment where they would be found in the mud. Yeah, so just to go over quickly, thermal cycling did not produce any spawns with these red mussels at this time. I think there's more research that can be done with the just the different aspects of the thermal cycling, like the temperature gradients and the length of time, to see if they would, if there is a preference there in some aspect. And the larvae, obviously, they were sensitive to competition from zooplankton likely, and that over the for the substrates, the bees was there was a lack of preference for the bees, while juvenile or conspecifics were preferred to juvenile mussels, which makes sense with the literature as well as their natural environment. And so the broader significance of this is like the ability to get have efficient methods of hatchery work done for rib mussels would be really beneficial for habitat restoration, particularly in this area of the Northeast. And it's an under-researched area that, and if we want to scale restoration efforts, it needs to be done in an efficient and a cheap manner. And so I want to make this a seamless transition from hatchery to the field work, and that's one aspect of why I chose just bees to test this substrate material. And I think it's supposed to be layered pretty significantly, and that's an aspect too. That wasn't as done as much in our project. Yeah, I want to thank NUPAC for funding, NEPAC, and Dr. Sclafani at the Cornell Cooperative Extension. Thank you so much. Couldn't have done this without you, as well as Demetrius, Amanda, and Stephen from Flax Pond. Thank you so much.
Thank you. All right. Hi, everyone. I'm Anna Meichenbaum, and this summer I worked with the Cornell Cooperative Extension to do an ecological study of Hippocampus erectus for the northern land seahorse in Shinnecock Bay, Long Island. And specifically, we utilized scientific and citizen science-based monitoring techniques. So assuming you guys are all like me and didn't know that much about seahorses before I did this project, I'm just gonna give you guys a quick little rundown. I think the most well-known fact is that the males get pregnant. They give birth to about 100 to 300 babies that are about 7 to 12 millimeters each. They give birth for a few months during the warmer spawning seasons. And they also form monogamous relationships throughout those seasons, meaning they go to the same partner, and it's also assumed that they go to that same partner throughout their entire life, which is pretty cool. And they do a courting ritual, which they do like a promenade dance side by side, and they also can exchange colors while courting. And then next, I like to call them the aquatic chimera because they have so many different characteristics that are not really seen in other underwater species, such as that downward pointed horse head. They have the kangaroo-like pouch used to hold their young, the prehensile monkey-like tail, and then of course the chameleon qualities such as changing colors, and they also have independently moving eyes, which they use to hunt. And speaking of camouflage, they camouflage into their habitat, changing colors, and they also do cryptic behaviors such as turning around in eelgrass, which makes them really hard to see. They all are also considered true fish though, because they have gills, vertebrae, and fins. And if you were to like, turn a fish up on its side, they look pretty similar. They also have a fused jaw, so they don't have teeth, but they take in their prey through a sucking motion. And they're also very poor swimmers, which is why they like living in eelgrass to use their holdfast blade. And then if this video works, we have a video taken from Cornell of a male seahorse actually giving birth. You can see their babies are almost like exact copies of themselves, of the older ones, and they can swim and eat by themselves like as soon as they're born. Okay, so now some more background on Hippocampus erectus. The northern line seahorse is the only seahorse species that is native to New York waters. They live in the eelgrass meadows, and it is thought that they have sight fidelity, meaning that they don't leave their home range of about 1 meter to 2 square meters. And because of this, they're considered to be indicator species of healthy habitats, and they can also be an ambassador for eelgrass restoration. One of the reasons this study is so important is because there are no studies of Hippocampus erectus in the wild. The only things we really know about them is when they're pulled up in trawls. We see that there's a seahorse there, but really they're only studied in captivity and tank studies. And then also they have a coronet on top of their head, which is like little tiny growths. And each one is individual to a seahorse, so we can use image J analysis to measure those coronets to test for individuality. So some more motivation for this project. When this species was studied last in 2016, they were placed as vulnerable on the IUCN Red List and are still on that list today. There are a lot of current threats to them worldwide, such as use in traditional Western medicines, and they're also used in like aquarium and curio trades. And then more localized, they're often caught in fishing nets and trawls. And a lot of people don't know it's actually illegal to capture seahorses and keep them if you don't have a proper permit. And then also if they're like captured in nets and trawls, if they're out of water for too long, they will die. And also just like being captured, they could become stressed out. And even if they're put back into the water, they could die. There's also been massive eelgrass depletion throughout all over, but also throughout New York. And according to the 2008 New York State Seagrass Task Force final report, about 89% of our seagrass has been depleted since the 1930s, and it has not come back. And then Shinnecock Bay was picked as our study site because of all the restorative efforts that are going on from Stony Brook University as well as Cornell Cooperative Extension. And the results from this study will help for those restorative efforts. So there's 3 main objectives for this study. The first being to monitor the Hippocampus erectus populations in the Shinnecock Bay. We're trying to determine if Hippocampus erectus actually does have sight fidelity in both natural and restored eelgrass meadows. And then finally, we want to share this information with the general public. So to complete the first objective, we use a variety of seining and scuba diving events. For our seines, we used 1 meter by 2 meter mesh PVC nets we attached zip ties to the end that kind of stuck out, which acted as the combs. When we dragged that through the eelgrass, they would separate the seahorse from their holdfast blade and scoop it up. We also use scuba diving, specifically the iSeahorse protocol, which is a huge global database for seahorses. But for that, we would swim side by side, making sure not to overlap with each other, and measured out where we were swimming through. It was pretty difficult though for scuba diving just because, especially on Western Shinnecock Bay, the visibility was very poor. So while scuba diving, we actually didn't see any seahorses. We all collected all of our seahorses through the seines. But then also I decided to do a seahorse and pipefish analysis because both of these species are sygnathids, so they're in the same family, meaning that they utilize the same habitat and also eat the same food. When I first started seining, we did not see any seahorses, and I was getting a little bit nervous that I wasn't going to get any data. So we started doing pipefish counts, and if pipefish are being found in that eelgrass meadow, it can be indicative that this meadow is healthy and viable enough for the seahorses to live. We just hadn't been seeing any. So that for the next objective, to determine if the seahorses had site fidelity, this was mainly done when we caught a seahorse. So when we found one, we would take a picture of it with a ruler in the frame, so we could go back and measure the exact length on image J, like I talked about. Before we let the seahorse go, we would drop a buoy where it went, so we didn't stay over that same area that day, and also so we could return to that same point in subsequent years. We used image J analysis to measure the length and the coordinates, as I said, just to see if we had pulled up the same one year after year. And then we looked at restored natural meadows in Tiana Bay, where Cornell has been doing some eelgrass restoration, and then natural meadows by Ponquag Bridge. And these two areas were chosen because they had seen seahorses here in previous years. And then the final objective was to share information with the public through a list of events like we did here, and also now to share all of my knowledge with you guys, so you guys can then go out and tell people about what you've learned today. So now on to our results. So after doing 5 seining events and 2 different dives, we found only a total of 13 seahorses, and they were only found at the southeast side of Ponquag Bridge in natural meadows. But for the pipefish, we found a total of 233 pipefish, 124 of those coming from Ponquag Bridge and 109 of those from Tiana Bay. So that's saying that even though we didn't find these seahorses at Tiana Bay, they have been seen there in previous years. So the seahorses still could be there, and we just didn't pull any up because they are viable ecosystems. So out of the 13 seahorses that we captured, 6 of those were male and 7 of those were female. All of the males were pregnant, so that means that there are at least 6 mating pairs in the area, which is pretty cool. And so I mapped the locations in GIS, and it's kind of cool to see that each of the males were found relatively close to a female seahorse, which probably means that those 2 are pairs. But it is also important to note that the female seahorses do travel further than that home range of about 1 meter to 2 square meters. And after doing the image analysis, none of the seahorses that we found were the ones that we had seen in previous years. So we were unable to determine if they had site fidelity, but now knowing where these seahorses are, we can go there in upcoming years just to retest to see if we do pull up any of the same ones again. And then finally, I plotted anecdotal sightings from the past 5 years with the sightings that we had seen this year, and just all around Long Island. And this kind of shows seahorse hotspots that we could visit, in coming years, just to see if those seahorses have site fidelity or not. So in conclusion, even though we did not see very many seahorses, the information that we did find is still important for population studies, and we found all of those seahorses at South Ponquag Bridge. And also our information aids to existing programs such as Cornell's Marine Meadows program, letting them know where to potentially plant more eelgrass, because there are those healthy, viable ecosystems where the seahorses are. We all are also adding to the iSeahorse database, which is that global database of all the seahorse sightings, which will let people elsewhere know where we have found seahorses. And I did want to mention our thoughts into why we think that the seahorses that we found were only at one area. And we think that could be because there's an eddy that forms at the southeast side of Ponquag Bridge. Like the water comes in through the Shinnecock Bay inlet, and it then may pull all the nutrients to that area and also bring in the small seahorse fry. And it just circles there. And why would the seahorses want to leave if there's food healthy eelgrass for them. Future studies could potentially be looking to see site fidelity over the years, but also we kind of want to look into where seahorses go in the colder winter months, because they're not in the eelgrass. They leave and go out to much deeper areas, but we're not really sure why. But some of the DEC trawls have pulled up seahorses in over 100+ feet of water, and we're kind of curious as to why that would be, because there's no hold fast there for them to hold on to, and they are very poor swimmers. So before I end, I just want to do a quick thank you to the following. To all the different seiners that we had come out and help us, specifically the Huntington Breast Cancer Action Coalition, and to all of the photographers at Cornell and at this program. And then finally, I want to thank the Cornell Cooperative Extension, especially the Habitat Restoration team, Steve, Kim, and Jason, who unfortunately couldn't be here today, as well as Maddie, my partner in crime over the summer. And I wanted to thank Stony Brook University, specifically Ellen, for being my advisor. And yeah, I have one last video of a seahorse that when we released, just swimming. Yeah, and that's it. Any questions? Oh my gosh.
Do you think it have anything to do with like pipefish and like how they occupy similar niches and the glass beds?
Yeah, so I don't think they like directly compete with each other.
Okay.
It's just, they're just in the same family, like ones, if they just compete with their living together.
So you don't think they like compete for space or anything? Because I noticed that you caught like a lot of pipefish versus seahorses.
I think they're different parts of the water column, because the seahorses are like mainly at the bottom, like at the root, at the whole fast, and the pipefish are more like at the top. But yeah, yes, go ahead.
So I don't know if you knew that there is like a pipeline that was going from the south side of Hong Kong Bridge. And they took that out this year, and it kind of just messed up the whole habitat, because last year there has been seahorse sightings over there.
Okay.
And also eelgrass. Do you think like that can cause an effect of your data? Like I saw some seahorses actually like under Hong Kong Bridge, which is not like a normal habitat because there's no eelgrass there and it's a lot of current.
Yeah, so the current is interesting, because they like holding on to things, because that current will just sweep them away. Do you know, is there like derelict Fier or anything?
Yeah, so there's like different types of they're like living like a pipe slash wood platform material that kind of like holds them off, and there's like things that they can like hold on to.
Yeah.
But it's just weird for me to see that, and I was wondering if it's like because of that pipeline that like caused a lot of disturbance in a way and confusion for the seahorses.
Yeah.
That, like they kind of dispersed.
Potentially.
I don't know.
Yeah. I think we're out of time.
Okay.
Hello, my name is Peter Martin, and I worked with Cornell Cooperative Extension 2 this summer and Project Vesta, who is a public group that researches carbon capture and ocean acidification. So the project I worked on was survival and development of horseshoe crab eggs exposed to olivine-derived nickel. So we know what horseshoe crabs are, but why study them? What's the motivation? Well, first, their eggs are an important food source for migratory birds like the red knot and different fish species too. And ultimately, this helps fuel the marine food chain. Next, adult horseshoe crabs are a bait source for whelk and eel. And lastly, their copper-based blue blood is used for medicine and vaccine testing by the biomedical industry. Now, on the other hand, ocean acidification is the gradual lowering of the pH across surface waters. So the ocean absorbs about 30% of all CO2 from the atmosphere, and it holds it in. And since the Industrial Revolution, a 0.1 decrease has occurred across surface ocean waters in pH. It might not sound like a lot, but this is logarithmic. So it's a 30% increase in acidity. And this impacts coral reefs, ecosystems, and a bunch of different species. So this project is formed by a partnership with Cornell Cooperative Extension, Stony Brook, and Vesta. And the goal is to assess any impacts of olivine on an intertidal and nearshore organism. So we chose horseshoe crabs. What's olivine? Well, it's a nature-based mineral that can sequester atmospheric carbon dioxide. So that being said, it has strong potential to capture carbon and reverse ocean acidity However, it does release trace metals like nickel and chromium while it weathers. Therefore, it's important to study how intertidal and nearshore organisms react to olivine, but in a marine, rather an experimental setting. So this is what olivine, well, you can't really tell on this, but this is in North Sea Southampton where Vesta worked last year. And the olivine has a green tint to it. Can't really see it in the photo, but I'll come back to this in a bit later. This is our experimental setup at Flax Pond Lab. So as you can see in the water bath, it has water that filters pretty constantly. We have 5 different trays of treatments. We have control, nickel 1, nickel 2, nickel 3, and nickel 4. These are all kept at a constant temperature of 22 degrees Celsius and a salinity of about 26, 27 parts per thousand. We had a schedule for our experiment. So day 1 is observations. We have 3 days. First day, observations, is assessing dishes for development and health. Day 2 is maintenance. So this is when we actually remove and record dead eggs. We rinse all the dishes with DI water and alcohol. We gently place the eggs back into the dishes with a new treatment that we created that day. This is day 2, like I mentioned. Now, the concentrations we use were all based in seawater. So control is just seawater. Nickel 1 has 15 micrograms per liter of nickel concentration. N2 has 30, and so on and so forth. Why do we choose these concentrations? Well, nickel 2 is what's actually experienced at Southampton from the olivine that's currently weathering right now. So we went above and below those concentrations to test our experiment. And the third day is sampling. So as you see, there's a lot of different Petri dishes for each treatment. So on every sample event, we would take 1 row of dishes. So 3 replicates from each treatment. So that's 15 total. We would count the dead eggs on the mortality data form that we use. And then we would do a representative subsample from each of those 15 dishes. We would identify eggs. Then afterwards, we would place the eggs that we subsample, the 15 dish eggs, into a falcon tube, bring them up to Dana Hall, up the hill, measure the wet weight, and then put them in the oven for 2 days. And after that's done, we measure it for dry weight. And eventually they're going to be sent to a mass spectrometry lab where we can actually find out how much nickel was taken in by the horseshoe crabs. Now, I mentioned that we identified a subsample of 10 eggs, and we did this based on Sekaguchi, who's a biologist, his horseshoe crab development landmarks and parameters. So he has 5 different categories. This is category 1. That egg is about 1 millimeter to 2 millimeters big. So they're very hard to see with the naked eye, but you could see some formations of development, some clusters, especially in that middle left photo. Typically, we had the top left and the right middle photo. That's usually what we saw. After that is category 2. So you have more concentrated development with some rings around it, as seen here. This is when it becomes easier. We have category 3. So that's when you can actually see the rows of legs and the arms, not arms, but just the legs developing. Makes it exponentially easier to identify at this point. Then category 4 is really fascinating. You can see the actual shell and the legs and the eyes on the outside, and they move around a bit in their shells. So here's a video of what that looks like under a microscope. And they're pretty top heavy, so they always end up on their legs, on their back rather. And you see the legs kind of moving around. And lastly, category 5 is when you see a well-formed shell with the eyes, and the legs are so long. They have little hairs that stick out of the shell. And here's a very unique video that we caught of a category 5 egg, trilobite larva, horseshoe crab, that's just about to hatch. You can see the opening of the embryo is actually broken. We didn't catch it actually hatching, but that's a really unique video, definitely. So after this, you have actual hatchlings. They're just really small horseshoe crabs, clear. Okay. And yeah, they're about half centimeter big. Here's one that's just learning how to walk. They're really speedy. That's how small they are too. Here's one moving its legs under a microscope. And like a full-size horseshoe crab, they contract, and then they kind of bend out a little bit. Here's some abnormalities we was out with, just discoloration, eggs that might have hatched too early. Here are the preliminary results. You have mortality on the left and the different sample days on the x-axis. This is average mortality versus time. So over time, we found that control had the lowest mortality and N2 had the highest. N1, N3, and N4 kind of met in the middle. Now, the inverse of this is average probability of survival. So control has the highest probability of survival, while nickel 2 has the lowest. And N1, N3, and N4 all meet in the middle too. Now, to find proportion of hatchlings by treatment, because counting all the hatchlings from the last sample day doesn't account for how many eggs actually started off in that dish. So this is calculated with both of those values considered. And you can see that N4 has the highest proportion of hatchlings based on how many eggs started off, with control and nickel 1 kind of holding the rear. Now, another way to assess development was to do morphometric analysis. So I used image J based on the hatchling photos we took. We took dorsal and ventral photos of the horseshoe crab trilobite, and we did the prosomal width and prosomal length. Prosoma is like the shell of the horseshoe crab. So we measured these based on the ruler in, we calibrated it with image J. And these are the results of that assessment. So we didn't find much of an effect on the average prosomal length or width of the hatchlings. As you can see, there's not much of a fluctuation, but it's still good to consider. It's good for us to know because you have to find out if it might have a possible effect. So overall, our preliminary results suggest that there might be a relationship between egg mortality and nickel treatments. Maybe there's an optimal range of nickel levels that exists for horseshoe crab development, because this experiment was about 1.5 months, 2 months long. So this might benefit eggs early on, like we saw, but overall, with cumulative exposure, this might impact survival. And future work, we are going to send away the experiment samples that we tubed at Dana Hall to a mass spectrometry lab to find out how much nickel was taken in. The nickel concentration was taken up by these horseshoe crab hatchlings, and then compare these results to the mortality and hatchling success that I just showed you. And lastly, we're going to analyze the field data because, as you can see in this picture, I'm on North Sea Beach in Southampton, which is where we had to complement the experiment sample, the experiment that we did at Flax Pond. We also worked at North Sea Beach. So we worked in 3 different sections: a control section, a section of low olivine, and a section of high olivine. The olivine is nourished; it's in a sand nourished this coast that you're seeing, so it enriches the sand. And in each of these 3 sections, we had 3 different holes containing 8 bags each that we prepared at Flax Pond. And this is to show us how the eggs develop in a natural setting with natural weathering of olivine, as opposed to what we did in the experiment, which was very controlled and with very specific concentrations used. And I would like to definitely acknowledge the invaluable and generous guidance from Dr. Sclafani and Dr. Serrato, who I couldn't have done this project without them. CCE, Project Vesta, Stony Brook, Flax Pond, all the help that I had, my coworkers and volunteers. There I am digging a hole at North Sea. Very happy. And that's all I have.
Okay, we do have time for questions.
Yes.
Aside for mortalities, did you keep track of which groups had the highest amount of egg abnormalities?
We took notes. We had these data sheets that we used, and we took notes anytime we saw an abnormality, but it wasn't much of it wasn't very significant, or else we really would have kept maybe even a separate data sheet for that. So there were many, but there weren't that many, perhaps.
Yeah, so obviously that's working in Southampton for a while, I know, so looking what you're doing now, did it change anything on how they're going to continue doing work in Southampton? I'm sure it's all in the picture now.
It might. This is kind of modeled off an experiment they did last year, which I can't quite talk about, but based on the results, be careful with what I say, based on the results of this experiment, that might encourage a future, another follow-up study for the 3rd year, next year. Because our experiment improved upon theirs from last year by adding Flax Pond. Last year, they just sampled at North Sea, Southampton. Yes.
So I have an overarching question for you as the world searches for a solution to our carbon problem and some of the bigger impacts of climate change, which I think is at the heart of this work. We're constantly looking for additions to the natural environment and highly engineered solutions. In this case, do you think that scaling up the use of olivine on coastlines worldwide, which is what Best has in this proposal on their website, is something that could have potential detrimental effects to fish species in the littoral zone? I mean, I know that's kind of why this work is being carried out, and I think it's great that you're a part of it. I'm curious, you know, you look at sample sizes and these certain areas, but if you think about multiplying this to the scale that they're looking to do this, do you think that could potentially have a deleterious effect?
I think it could, and that's why it has to be researched on a bunch of different organisms, especially not just from here, because we used Atlantic horseshoe crabs, Limulus polyphemus, but there's 3 other species of horseshoe crabs in like the Indo-Pacific area. So, but aside from just that species, it's important to see how this impacts just the ecosystem as a whole. I don't think anything is going to be a perfect fit, but definitely have to find out, what kind of bad impacts it has, bad negative effects.
So
hi everybody, my name is Kate Alexander. I am an MCP part-time student, which is probably why a lot of you don't know me. I'm just sharing my screen. And I was interning at Mangrove Action Project this spring. All right. Yeah, so I'm interning with Mangrove Action Project. I am doing a pre-recorded video because basically I'm graduating in the spring, and so it doesn't make sense to do it in August, but we'll be playing this in August and I will be there via Zoom to answer questions afterwards. So my project is called Investigation of the Hydrologic Fishbone Method, case study of India's unquestioned mangrove restoration model in Andhra Pradesh. Andhra Pradesh is a state in India. The Mangrove Action Project is where I intern. They're super cool. It's based in the US, but it's a nonprofit that travels around the world for mangrove restoration. So mangrove restoration usually looks like a plantation where a bunch of canals are built, and next to those canals, mangroves are planted. Community-based ecological mangrove restoration is what my organization does. They don't actually plant any mangroves. They find out where mangrove degraded areas are, go and visit those places, talk to the community, find out where the waterways used to exist when the mangroves were healthy, make sure that there's no marine pollution issues in the area, and then have the community go and actually dig out and restore the hydrology that used to exist there. And then naturally mangrove seeds come with the tides, and they're dropped from their trees, and then the tides will naturally bring the seeds in to the area that was hydrologically restored. And those mangroves, the natural species of the area will grow, and the mangrove forest will become reforested over time, which is the best ecologically friendly, it's just the best way to do it. It works the best because it's just how nature works, and so it's really cool. But I'm going to be talking today about mangrove plantations. So the fishbone method is a mangrove restoration method used in India. Their forest department is mass implementing it in the states of Andhra Pradesh, Tamil Nadu, and Odisha. The motivation for this is that mangroves sequester huge amounts of carbon. The climate has way too much carbon dioxide in it right now. We need to make sure that more carbon does not, get into our atmosphere, and so sequestering carbon via mangroves seems like a very good way to also fight sea level rise, storm surge issues. Protecting mangroves are so important. They're a nursery habitat. they do so many different things. I just don't have time to talk about that. So I have 10 to 12 minutes to present. And I have a lot to say about my thing. So yeah, I love snorkeling with mangroves. Basically my organization brought me on as a research intern to study the fishbone method, which is that mangrove plantation restoration method that they are implementing all over India. So they wanted me to look on Google Earth, look at the different imagery that they have from different periods of time, from satellite swaths over the years, and they wanted me to see, what's going on with those sites over time. Is the mangrove cover like doing well and growing and then dying off later? My organization is really just worried that mangrove leaves, sediment, basically mangrove leaves fall and then they decompose. They get stuck in the canals next to the mangroves in these plantations. And they're worried that without someone coming in periodically to dredge and dig out these canals, the canals will get clogged and then the water will be able to get to the mangroves, and then the mangrove sites over time, as the mangrove leaves fall, when they get clogged, the mangroves that were planted will eventually die. So it's my job to find out more about the fishbone method in general, via scientific literature, and then to find out whether or not many years into the future the fishbone method works without human intervention and dredging. So I had to do a scientific literature review because that's what they had wanted me to do, but I looked through scientific literature. I spent so much time looking through the peer-reviewed literature, and I found only 4 peer-reviewed articles mentioning fishbone. 2 of them, well, one of them, like, barely discussed fishbone at all, just mentioned it. The other one was extremely useful. Then there were 2 that you had to pay to be able to read, and I wasn't going to spend $60 to read one of them. So I really only read 2 peer-reviewed sources after spending like 2 weeks looking for peer-reviewed sources. So there's a huge empty space where a bunch of peer-reviewed literature could be and should be. Then I looked at the satellite imagery. That was the second half of my. But yeah, I ended up having to do a literature synthesis instead of a review, because writing a scientific literature review when there's not a lot of peer review stuff, but most of the literature is gray literature that's just like manuals, and they're all saying the same thing. They're all being published from the same groups. It just isn't interesting and it's not helpful to my organization. So they were like, yeah, okay, these are the results. Basically write a synthesis of everything that you've learned instead of reviewing the literature in a scientific literature review. Then the second half of my internship was me studying the satellite imagery over time. So I had to quantify, the success of, or partial success or failure of each site, fishbone site that I was looking at. I conducted 3 case studies of mangrove forest areas within Andhra Pradesh's Godavari River Delta. Each of the 3 places are pretty much right next to each other along the coast, and they're attached to tributaries of the Godavari River coming down along the central east coast of India. So the forest areas are Belusupipa, Kovapalam, and Kraemer Wildlife Sanctuary. I apologize if anybody in the crowd is from India and I've mispronounced that. I just don't know anybody on the ground that could tell me, and I'm looking at satellite imagery, and I'm just sorry. I wish I knew how to say them. But yeah, so the literature synthesis, lack of peer-reviewed sources. Generally, the research on fishbone was very minimal, and the only organizations even publishing anything about it are basically the organization that was founded by the man that created fishbone, the MS Swaminathan Research Foundation. MS Swaminathan basically invented fishbone and started mass implementing it. And then the Forest Department, India's Forest Department, they're the chief implementer of the fishbone method in India, and they're getting tons of funding to be able to do it. So both of those organizations have bias and are getting something out of the fishbone method doing well, or at least being mass implemented. So really, independent research from organizations not attached to those groups is extremely important and lacking. The investigation of satellite imagery, basically I had to define what success, partial success, and unsuccess was for methodology. I chose only sites that were 10 or more years old. The successful site was, I decided it would be greater than 75% mangrove cover of the site area being sustained over time, over 10 years. Partial success would be the growing and sustaining of mangrove cover, 35 to 75% cover of the site, 10 plus years in the future. And then unsuccessful was the same, but less than 35% mangrove cover sustained. You're probably looking at my screen, but I'm sorry, I'm taking my sweater up, I'm a little warm. Okay, so the case study, the first case study that we're looking at is SoMAS. There were 9 total sites. 3 of them were successful, 6 were partially successful, and none were unsuccessful. That is really just not what my organization was expecting at all. This is what fishbone looks like. The fishbone is, I don't know if you can see my mouse, but there's a main canal that's 90 degrees from the regular natural waterway. It's the thickest in the main canal is much wider than the others. Then 30 degrees off of that main canal are the smaller canals, which mangroves are planted on either side. And so it looks like a fish's skeleton. On the right, you can see an image. It's from the left image is 2009, the right image is 2022. You can see that many years later in this site area in Velasquez, it's partially successful because the mangroves are growing, you know, around the smaller canals, but not spreading past that. Then here's another example of a fishbone site at Velasquez. This is a fully successful site. On the left, 2009, on the right, 2022. The right image is basically completely covered in mangrove cover, so mangrove brush, and that's really cool. It's very exciting. It's just really not what we were expecting at all. A second case study. This was the least successful of the 3 case studies, but still pretty successful. There were 13 total sites that I had reviewed in Coastal Column. 2 of the 6 sites were successful. 9 sites were partially successful, and only 2 sites were unsuccessful. So out of 13 total sites, only 2 were unsuccessful, which is just really, again, not what we were expecting at all. This is a partially successful site. On the left, 2005, on the right, 2022. And again, you just see the mangroves not spreading past the canals that they're at. Here, this is a fishbone site 7 at this was a fully successful site. And on the left is, I believe, 2009, and on the right is 2022. On the right, just in the area that where the canals were actually placed, it was greater than 75% mangrove cover sustained. So this is successful site. That's what one looks like. And then not fully successful, but it is successful according to my methodology. And it looks like it has really done a good job of reforesting the area. A lot of these sites are old aquaculture ponds that failed for shrimp farming and prawn farming, things like that. That's a big mangrove degradation issue in the area, and so the forest department is trying to reforest the degraded areas. So here, this is an unsuccessful site. On the left, it's a huge site, it's really big, and then all the way up on the right, so 2009 on the left, 2022 on the right, it's just a lot of it is left there. It's less than 35% cover sustained. Then our third case study, I found like 28 plus sites here, but I just didn't have the time to actually review all of them and keep the satellite imagery and find all of the information on it, etc. It just takes time that I didn't have. So I only was able to do 3 total sites here, which I'm really sad about because there are just so many, and I really love this project. But so the sample size was small here, but of 3 total sites. All of them were completely successful, greater than 75%. Mangrove cover sustained over 10 years, which is incredible. Really just not at all what we were expecting. The thing is, this is all 3 case study sites are within the Godavari River Delta, and they're mangrove forests in like Delta mangrove forest, which is like a type of mangrove forest. And so this is just representing success in that specific area for that type of forest. And we still don't know if these sites are being dredged periodically, and that's why they're succeeding. That's something that I could find out if I went there and I started talking to the local people and things like that. Something that would be great if I was able to get funding for this project to continue on. It's just very valuable information, and you know, for millions and millions of dollars to be flushed into the forest department when they're not creating something that's long-term sustainable and self-sustaining. It's just, it needs to be researched. Also, the areas, so this is the success of the different, I just made a figure showing that. I thought I'd have more time, so I was gonna talk more about that, but basically 60% partial success, 32% full success, only 8% of all sites unsuccessful. Amazing. Basically, we need more peer-reviewed sources. The Indian states of Tamil Nadu and Odisha need studying because these states are well known as places where fishbone has been very unsuccessful. We had expected to see less success in Andhra Pradesh just because we knew bad things were happening with fishbone in Tamil Nadu and Odisha. And it was another place, Andhra Pradesh was a place that they were also implementing the fishbone method, but basically these Tamil Nadu and Odisha I haven't even looked at yet and studied. They're well known as not having good fishbone success. And we need to know why there it was unsuccessful, why here it has been successful, the factors that go into that, etc. So future research, there's tons to learn. And yeah, we need independent groups other than the MSSRF and the Forest Department actually studying all of this. So this is my works cited. And I have a bunch of them. A lot of them are just coordinates on Google Earth Pro, but eventually it will say questions. Does anybody have questions? And this is my email if you ever want to email me about any of this. And I have like a marine Instagram where I just like post stuff about marine life. And if you'd like to follow me on that, or if you talk there, that works too. But thank you, and I will take questions now.
All right. Hi, everyone. My name is Katie Miller, and the title of my project is An Assessment of Diversity and Equity in Youth Environmental and Marine Education in Western Suffolk County, New York. So I completed my internship with Cornell Cooperative Extension of Suffolk County Marine Program. I am a natural resource assistant educator for them. So I create, edit, and present educational materials to youth programs, schools, and community events across the Northwest Suffolk County region. And during the summer, I assisted with all aspects of our Sea Stars Marine Day Camp that we run. So here's just some photos of some of the programs that we do. So this is a touch tank. This is me teaching a lesson about horseshoe crabs. This is our Stormwater Superheroes program. So this demonstrates the effects of stormwater runoff into our waterways on Long Island. And here's just some photos from the Sea Stars camp that we run, teaching a lesson, and then the children on their daily nature walk. I think they were fascinated by a caterpillar in that photo. So getting into my actual project, environmental education, especially of youth, is without a doubt one of the most important aspects of environmental behavior and attitudes. And this includes marine education, which can lead to ocean literacy or understanding how the ocean works. However, in the US, low levels of ocean literacy have been identified, and this acts as a barrier to engage in positive environmental behavior and to consider marine-related careers. Further,
environmental education contributes to this idea of a sense of place, or the bonds and attachments someone develops in a particular location. And sense of place has been shown to increase positive environmental attitudes in youth in rural and urban environments. So I'm sure many here are aware, we are surrounded by incredible marine life. But many people who actually live on the island who aren't in this field are unaware of this. So I believe that environmental education can really help to make positive environmental change on Long Island through not only the transfer of knowledge, but also through the establishment of a sense of place on the island. And finally, environmental education encourages young people to pursue environmental and marine related careers. And that leads me to my next point. The environmental and marine fields are among the least diverse. People of color make up somewhere around 43% of the US population, and only 16% of the staff in environmental not-for-profits, government agencies, and grant foundations. And this continues throughout the environmental, marine, and conservation fields. So low diversity in the conservation fields limits the fields in many ways. For one, people of color and other traditionally underrepresented groups are the most affected by environmental issues and the impacts of climate change. So low diversity reestablishes cultural biases when creating policy and implementing management. Low diversity also limits the opportunity for cross-cultural knowledge exchange and innovative policy and effective management and conservation for marine and environmental. So for my project, what I wanted to do was analyze the racial and economic demographics of the public elementary and middle schools that CCE Marine program has and has not visited in our Western Suffolk County regions from 2005 to 2022. I wanted to determine if any trends, patterns, or changes exist in these demographics over that 18-year period. So this is the study area. These are the northwest in orange and southwest in purple CCE regions of Suffolk County. Thank you so much to Mike, our GIS specialist, for creating this map for me. So this is where the public school data came from. Briefly, so data was collected from our CCE past outreach documentation dating back to 2005. And our public school racial and economic data were obtained from the state's Education Department Information and Reporting Services Enrollment Data Archive. It's a mouthful. But racial demographic documentation began in 2005. And then the state only began documenting the number of economically disadvantaged students in 2012. So that's when that data began for us as well. And we used SPSS 2.8 software for the statistical analysis. And just some important definitions on the bottom here. Students of color are defined as those students who are not white, and economically disadvantaged students are defined by the state as those who participate in or whose families participate in economic assistance programs. So this could be SNAP or free or reduced lunch programs. So getting into some of our results, figure 1A shows the percentage of students of color in those schools visited by CCE Marine from 2005 to 2022. And figure 1B shows the percentage of economically disadvantaged students in schools visited by CCE from 2012 to 2022. So overall, neither the percentage of students of color or the percentage of economically disadvantaged students changed significantly over the two time periods. However, one key takeaway from these over time graphs is that the percentage or the amount of outreach that CCE performed in a given year was positively correlated with the percentage of students of color who were reached by CCE. So this is just emphasizing that it's important that we do as much outreach as possible in a given year to make sure we're reaching those schools with higher percentages of students of color. Getting into our comparisons. So this is the comparison of the percentage of students of color in schools not visited on the left and then visited on the right by CCE from 2005 to 2022. So
there was no significant difference in these two numbers. However, there were 5% more students of color in those schools that CCE did not reach in the 18-year period. And similarly here, this is the comparison of the percentage of economically disadvantaged students. So there was 5% more economically disadvantaged students in those schools not visited by CCE. However, this was not a significant difference. And a significant finding of this project was that there was a significant correlation between the percentage of economically disadvantaged students and the percentage of students of color. And this is in agreement with many other studies in the state and across the nation, which is why there was about 5% more economically disadvantaged students and students of color in those schools that we did not reach. So here we have the schools that we visited, did not visit, and all the schools in the Northwest, Southwest Suffolk County regions based on their United States Education Department economic category. So as you can see here, these pie charts are very similar. So most schools fall into the low poverty in yellow or the mid-low poverty in gray categories. So this is just demonstrating that not much economic variation exists in these regions. However, that's not to say that CCE shouldn't be doing more to reach more of these schools with higher percentages of economically disadvantaged students. As you can see, we only visited 5 schools classified as high poverty throughout the entire time period, but there are only 24 schools classified as high poverty in the entire region. So that's demonstrating that. And then below here, very similarly, the average percentage of students of color in the schools visited by CCE was about 30% over the entire time period. And the average percent of people of color in the 4 towns in Western Suffolk County, oh, you can't see that. Was 32% in 2022. So very similar numbers, just demonstrating that CCE Marine is visiting schools whose racial and economic demographics represent those of the regions that they are in. So like I said, we're reaching schools whose demographics reflect the Western Suffolk region, which shows little diversity in economic and racial demographics. However, more can certainly be done to reach those schools with higher percentages of students of color and economically disadvantaged students, especially since these two often coexist. And now CCE Marine can prioritize these schools with our advertisement and our outreach booking. And we can also advertise or prioritize those schools that we haven't reached in the entire 18-year period, because we did only reach about 69 schools, and there are 212 total schools that we could reach. So this should allow CCE to reach a wider range of students in Western Suffolk, helping to grow pro-environmental attitudes through not only knowledge, but also through establishing a sense of place on the island, and to encourage young people, and hopefully from those traditionally underrepresented groups, to pursue careers in the environmental, marine, and conservation fields. A huge thank you to Dr. Christine Gilbert. She was my project mentor. I could not have done this without her, seriously. She ran all the statistical analysis for me. Dr. David Taylor was my other mentor. Thank you to him. Carolyn Sikowski is my internship supervisor. She has supported me from the beginning with this project, so thanks, Carolyn. And Mike Sakulus, who is our GIS specialist. So thank you so much to all of them.
One quick question. What was your favorite part about your internship?
I mean, the kids, obviously. The camp that we ran was super, super fun. So definitely the summer camp. I felt like I was a camper at the time. So
Okay, thanks very much.
Okay. Hello, my name is Isaac Nikolic, and welcome to my presentation on a story map for homeowners on when to consider a living shoreline. Can everyone hear me all right?
No.
Am I speaking?
Let's move it quickly, like that.
How about now? Is that better?
Much better.
Okay, so I'm going to talk to you about my process for choosing this project, my research and findings, and finally the story map that I'm creating for the Ecological Estuary Partnership. So first, how do we get here? I've had a personal interest in living shorelines since 2018 when I wrote a research paper on their benefits for my Writing 102 class. So when Dr. Joyce Novak came to Ellen's capstone class to present on fellowship opportunities and mentioned living shorelines, I was excited to apply my interest to a real project. For the first few months of fellowship, I helped to introduce Dr. Tara Ryder's SUS 401 class to living shorelines so they could research recommendations for changes to permits in the Peconic Estuary townships. Then the focus changed to my own project, creating a story map to help homeowners understand the environmental variables involved in deciding if a living shoreline is the right kind of protection for their homes. This project is part of the work of the Shoreline Adaptation Initiative and Work Group run by the Peconic Estuary Partnership and the New York Sea Grant. Dr. Kathleen Fallon of New York Sea Grant has been essential to its progress. So a little bit of information about PEP. PEP is a national estuary program, which is an EPA place-based program to protect and restore water quality and ecological integrity of estuaries of national significance. It's a coordinating organization on Long Island, working with many partners to fulfill its objectives. For example, they recently worked with Cornell Cooperative Extensions, Suffolk County, and the town of Southold to create a living shoreline demonstration project at the Suffolk County Marine Environmental Learning Center. So what is the need for my project? PEP has received questions recently from homeowners who have tried to apply for living shorelines and run into problems. They had trouble understanding the permitting process, what information they were responsible for filling out themselves, and how they could understand the information needed to decide on a shoreline type. New York Sea Grant is already working on a permitting guide for coastal projects, but there is still much room for education on the physical side of the issue, how to choose a living shoreline type and the information that goes into that decision, and that's really where my work comes in. So to start that process, first we have to know what living shorelines are. And the name can be a little bit confusing because it goes by many other names, green protection, soft protection, etc. And living shorelines as a title is a little bit confusing because they're basically made of living and non-living components, but that's just a kind of buzzword that tends to stick with people. So it's a form of coastal protection made of natural and constructed materials, such as marsh grass, organic coir mesh, rocks, concrete, and oyster reefs. And the goal is to reduce the risk of erosion or storm damage to a shoreline. Examples include edging, which hugs fringe vegetation to protect it from wave energy, sills, which sit parallel to the shore and reduce wave energy, while also being capable of building up sediment on their landward side, and breakwaters, which are made of larger, more durable barriers than sills and sit further offshore to intersect larger wave energy. Living shorelines contrast hard protection like seawalls by not isolating habitat on their landward and seaward side, and by the ecosystem services that they can provide to an area. So now I'm going to give an example of why it's important to understand how the variables at a site are going to impact the living shoreline design you should have by showing a paper which didn't correctly take these factors into consideration. The top right image is from an article in the Journal of Environmental Management, Do Small-Scale Salt Marsh Planting Living Shoreline Projects Enhance Coastal Functionality, a case study in the Northern Gulf of Mexico by Amato et al. In 2022. So they did a good job of studying important variables at their sites, including erosion history and tidal variance, but chose poor materials and designs for a site experiencing modern to high boat wake energy with a high erosion rate. So for example, they used black needle rush as their vegetation when Spartina alterniflora has been shown to be better at handling boat wake energy. And they used coir logs and coir mesh as their protection, which are on the softer side of materials, even though the site was experiencing high erosion and high wake energy from boats. So the graph on the bottom right there is an example of a paper in 2015 by Manis et al., which tested the ability of Spartina alterniflora to reduce wave energy in a wave tank simulating boat wakes. And one of its main conclusions was that Spartina alterniflora, when paired with oyster reef, can reduce boat wake energy by over 67%. Something that they got right, however, was that they pointed out there is less research into the impacts of small-scale residential living shorelines compared to larger restoration efforts. Because of this and the modern push by some government sources like NOAA or the New York Department of Environmental Conservation to use living shorelines as a replacement for traditional protection, there is a potential problem where there isn't enough emphasis on understanding site conditions and matching them to effective project designs before a decision is made. So we need more research and we need less generalizations. So before I go into some of my findings on what will allow a living shoreline to be successful, depending on site conditions, I'm just going to define some of the important variables in that process. As previously mentioned, erosion history is vital to living shoreline success. Get this wrong and your project will be washed away before it has time to establish. Shoreline slope is the change in shoreline elevation from the mean lower low water line to the spring high water line. Marsh sills and oyster reefs require smaller slopes than revetments and breakwaters, but any project protecting marsh vegetation will do better on a shallower slope. Tidal range is the vertical difference between high and low water levels during a tidal cycle. Tidal range is important to marsh vegetation because it affects inundation, and inundation time will influence what kinds of marsh grass will do well at a site. Tidal range will also play a role on deciding a structure's crest height, because a breakwater becomes less effective at reducing wave energy the closer the water level is to the crest. Crest is the upper elevation of the protected part of a shoreline project, such as a rock sill or breakwater or revetment. Freeboard is another critical variable due to sea level rise. It's basically a margin of error based on incident wave height. Incident wave height is the water level that's expected in a rare once in 100-year storm, and the freeboard is how high above that level a protective structure should be built. Now I will share some of my findings for the appropriate site conditions for 4 kinds of living shoreline projects, which are included in my story map. Marsh sills are appropriate for sheltered intertidal areas such as estuaries or bays. They need at least 6 hours a day of sunlight for the vegetation to grow. The vegetation without protection can withstand current or stream velocities of 3 to 6 feet per second, but when protected by a rock or oyster sill, can withstand velocities of 10 to 18 feet per second. Although marsh sills can have shoreline slopes ranging from 1 to 15 to 1 to 5, the optimal slope for marsh vegetation is between 1 to 8 and 1 to 10. This is because marsh vegetation, while resistant to inundation, still requires a way to drain water from itself or it will drown. Related to this requirement is that the site have less than 100 square feet of standing water. Marsh sills can protect against wakes of up to 3 feet and a tidal range of 0 to 4 feet. Age 20% is the height that wind waves in an area will surpass 20% of the time. For a marsh, the age 20% value should be between 0.5 and 1 feet. So this is what the protective sill must be able to reduce the wind wave height to. Now moving on to breakwaters. Breakwaters, in addition to being useful in low energy areas, can also be used in more exposed areas like shorelines directly facing ocean conditions. Steeper slopes make vegetation more vulnerable to erosion damage, especially during storms, but because breakwaters offer greater protection than a sill, they can be used in sites with greater slopes. Breakwaters can also provide protection against higher erosion rates than a sill. Basically, they've been used in environments that have higher rates of erosion and more wave energy than any environment you're likely to use a living shoreline in. And so they're not really given limitations in living shoreline guides for what kind of environments they can be put in terms of wave energy or erosion rate. One of their significant limitations is, however, their soil bearing capacity because of the physical size and weight of the structures. So that has to be over 1,500 pounds per square foot, which is the highest of any project. Breakwaters are also the most expensive living shoreline, costing potentially thousands of dollars per foot to install and have more complicated permits. For the sake of time, I'm going to move on a little bit to the story map. So the story map that I'm making, the process of outlining it was the most challenging part and took the most time to complete. I had to balance technical information useful enough to be helpful for homeowners while also being straightforward enough not to alienate them. And I had to make sure that I was creating not just an educational document, but a tool that homeowners and governments could use to choose locations for living shorelines. The story map works like this. First, a user learns what to pay attention to in the story map. Then they learn basic information about living shorelines. then they're introduced to variables involved in evaluating site compatibility, which involves some of those diagrams that those pictures that I created before, plus additional information about variables and some useful tips. And then they're shown GIS maps, which can tell them information about their particular sites. They can learn about their flooding history, wind wakes, tidal vegetation type, shoreline slope, etc. And then finally, they're given advice on permitting and shown links to learn more about any particular subject of the story map. So the current state of the story map is that it is in the review and editing process by New York Sea Grant and PEP. Once those changes have been made, it can be presented to homeowners and policymakers on Long Island. Besides those next steps, there is room for my work to continue with the shoreline adaptation initiative. Basically, identifying gaps, more gaps in living shoreline guidance resources, and advocating for more demonstration projects on Long Island so that New York can get its own version of data and guides and engineering practices for creating living shorelines, as right now, most of those sources are from other states and not for our particular area. As professionals in the field of marine conservation and policy, it's our responsibility to provide the public with data-based education and outreach. With this story map, I hope to accomplish just that. I would like to say thank you to Dr. Joyce Novak and the Peconic Estuary Partnership and New York Sea Grant for this fellowship and the opportunity to work with you, Dr. Kathleen Fallon and Jade Blunow for helping me navigate the process of researching living shorelines and outlining my story map, Dr. David Taylor for helping me plan this project's timeline and giving me advice on time management and project priorities, and the marine and conservation policy for the memories that we've made together this year. Thank you.
Okay, there's no, not that button.
Oh, not that one. My bad.
That's all right. It's all
Sorry.
right.
I was trying to just get to the.
Yeah, let me help you get to that. Hang on. Just share the screen.
During questions, I have a draft of the story map that's complete, and I was able to log in so that I can show it to you during questions if anyone has questions about the story map.
I guess we don't have as much time as I thought, but we do have time for like one question. No question. Can you show us the?
Yeah, this is the draft. This is a draft of the story map, right? So the way that it works is I use a lot of sidecars. So I have an introduction section on how homeowners are supposed to use it, what they're supposed to be paying attention to, like I said in my presentation. Then I have the introduction section. I talk about what living shorelines are, how the government defines them, and then what sites they're appropriate for in general before I get into more specifics. Then I have a section on the types of protection, defining them, pictures describing them, the kinds of materials that can be used, links that describe those materials in more detail, etc. And then I move into kind of trying to understand the variables that are involved in the living shoreline process. Many of the images that I've already shown you during this presentation, defining those variables. The next section is what I call thresholds, which has a lot of those images that you've seen, plus a lot more information that I was trying to get through, but didn't have enough time for in the presentation. And then it moves into other tips and then goes to permitting, because there's a lot of information that's complicated for people to understand, like the joint application forms from the U.S. Army Corps of Engineers and the DEC, as well as how to find information about the tax codes and where they are, the tax map, et cetera, et cetera. And then lastly, I move on to the maps, where I have just maps that I've found throughout my research, such as one that helps you create environmental impact statements when you're filling out forms, one that helps you to understand the different kinds of variables that are out at your site, such as the wind wake energy, or if you're looking for the types of vegetation that grow in your area, et cetera, et cetera. And then at the very end, I have a list of all of the resources that were used in creating the story map for further review, yeah, et cetera. And I've probably taken up way too much time.
No, you actually, 20 seconds left, so you did perfect.
Great job. Can you just remind us, how do we find this story map?
So it's in the editing phase right now. So once it's published, it's going to be similar to other story maps that like Sea Grant or other organizations have made, and then it will be accessible to the public. But that's still a few months away.
Would that be through the Peconic Estuary Program website, or would it be
not
standalone?
Not sure yet. It could be from the Sea Grant or PEP. We're not sure on those details yet.
Okay, well, looking forward to seeing that be published too. Thank you.
Hello. Can everybody hear me all right?
Yes.
So for those that don't know me, my name is Tyler Potter. And this summer I worked under Dr. Donovan Finn, studying the economic impact of recreational saltwater fishing in New York State waters, sponsored by the DEC. And so this is my project, investigating the economic benefits of recreational saltwater fishing on the state economy, on state economics, in New York State study. Right, so the purpose of this study is to, still is to determine the economic and behavioral impacts of recreational and marine angling in New York State through public data analysis, online surveys, and focus groups with New York State residents who participate in recreational marine fishing. And so specifically, we want to better understand the behavior, choices, preferences, and again, economic impact of recreational fishers on the greater New York State economy. And so summer tasks, things I pretty much was in charge of, had to do this summer was conduct a detailed literature review of academic research on economic impacts of recreational fishing, review similar studies in all other coastal and Great Lakes states, I took a few SBU required online training to work with human subjects data, because in this project, so far we worked with survey data. I assisted in the cleanup of said survey data and conducted preliminary analysis of survey responses. And so this is essentially just a map of the quote unquote study area. Although some of the data we work with may come from fishers who are licensed in the state who come from out of state. But essentially, this is the New York Marine and Coastal District. It encapsulates Long Island, New York City, and parts of southern New York State above the city. And so, to begin this project, I kind of started with a literature review on so like fishing regulations, as well as various state reports and studies. But I just put this up here as kind of just to show what type of stuff that I was looking at, and just to reference, like, this is essentially the basics of New York State marine, like, recreational regulations. Most of the time, regulations are based on either the species or the type of fish, and then most of the regulations have to do with like minimum size, bag limits, seasons, and then every now and then you might have like big management things like closures or moratoriums. But yeah, and then pretty much rules like these are in effect for the New York Marine Coastal District, like I said, Long Island, New York City, Southern New York. And so recreational marine fishing as an economic powerhouse. So basically, whatever level you look at, whether it's local municipality, state, or federal level, recreational marine fishing, even just as a leisure activity, generates a lot of money, right? And money generates further economic movement, right? So for example, in New York State in 2020, recreational fishers took a total of 14,841 trips, generated 428 million in sales revenue, 189 million in income, it's hard to see, but 228 million in value added impacts, and supported 4,872 jobs. So just in case anyone's curious about what value added is, it's kind of like how somebody takes, you could sell a raw fish that you just caught, or you could take that fish and you could sell it to a restaurant, and then they'll sell a much smaller portion for how much they want. They added value onto the product. And then also what's kind of interesting is, this is just straight from the DEC website, for like fishing license holders. In 2021, 2022, there was about 900,000. fishing license holders, and if you kind of see it, it goes up and down. Mainly it's kind of plateaued around the 800 to 900,000 range. This is kind of consistent with like a national standard grid. Since about 2011, recreational fishing participation has been plateaued around 11% nationally. But if you look maybe 20, 30 years, it has actually gone down steadily a few percent. And then so willingness to pay, this is something that came up a lot in my literature review. And it's basically willingness to pay represents a fundamental aspect of like supply and demand. And in regards to fisheries, it's literally how much recreational fishers are willing to pay, willing to expend to participate in this recreation in fishing, right? So, and what that kind of means in regards to recreational fishers is how much are they willing to pay to literally get to their fishing spot, right? And then so if you're a manager, this kind of produces questions, right? So will they get more value fishing somewhere else? Are they willing to pay increased travel costs due to a closure somewhere for whatever reason? What is the economic impact of that closure onto the local economy or the state economy, depending on the importance of that fishery? And then so as this creates an incentive for fisheries managers to provide not only a quality fishing experience, right? So they want to catch good-looking fish, they want to catch enough fish, but also they also want to maintain the sustainability of the ecosystem in itself, because that's what managers are here to do. They're not here to close everything. They're here to find that balance between social and economic stability or whatever, and then also the environmental aspect. They really need to keep both going at the same time. So again, if fishing or environmental quality goes up, then usually participation will go up. Net willingness to pay will go up, will be more elastic if you're familiar with economics. On the flip side, if the water quality is poor or the fish are sickly, net willingness to pay decreases, and fishers go somewhere else. Your state or your local area misses out on that much money. So it's these types of questions and like behavioral aspects of recreational fishers that are really hard to model, and that's why the DEC wants this type of data. And then so just some interesting patterns overall across like various other states, because many other states, especially in the Carolinas and the Great Lakes states, have been producing this type of data in the past like 5-10 years. But like overall, there's like fishers are broken up into 3 basic categories, like shore fishers, private boating fishers, and charter party boat fishers. And there's pretty much a consistent hierarchy of expenditure. Like shore fishers spend less per trip, private boaters, excluding that initial boating cost, spend about the middle, and charter party boat fishers always spend pretty much the greatest, because they're not only paying for equipment, but they're also paying for that experience on a party boat and like the actual staff that's with them, right? So like other things, residential, like state resident fishers usually expend less per trip than non-state residential fishers, right? And that kind of has to do with the fact that people coming from out of state don't come as often and are usually more likely to charter or use a party boat for the service. Marine fishers also usually spend on average more per trip and annually than freshwater fishers. And just overall understanding these types of like patterns and how like your specific community or your management responsible area, what type of people they're comprised of, could really make a big impact on how you plan like management into the future. Right. So initial data collection for a project started with a survey. The DEC provided us a list of 10,000 randomly selected recreational marine license holders. And we conducted the survey through Qualtrics. And for this project, the expected response rate and the quota or goal was 5%, or about 500 return surveys. And then the goal was met, 537 surveys were returned. This is kind of it's kind of difficult to see, but this is kind of how the survey looks. This pretty much was multiple choice. Some short answer later, or partly introduced, but so we don't have exactly the results yet, but we do have data from the survey that we have the kind of preliminary analysis for. So I picked out like a few interesting data points from the survey. So like just characteristics, the ages of respondents skewed higher. So the largest group of respondents were above 65 or above. And there's another question that corresponded to this one that asks for employment. And the largest group of respondents, well, actually, no, the second largest group of respondents after employed was actually also retirees. So we're already seeing an interesting pattern with who was responding to our survey, right? And there was roughly a 50-50 split regarding the purpose of fishing, whether it was just for recreation or just for food. Majority of respondents were white. The most common education level was a bachelor's degree. This was probably the most interesting data that I saw, which was the household income. The responses to question 37 were, what is your total annual household income? So the most common answer was the most wealthy group, over $150,000 and above. I thought that was very interesting. The rest of the graph pretty much for the bell curve. I will say though that the second highest group, almost 20% of people preferred not to answer this question. This was the highest. There's always people that didn't answer questions, but not to like the extent of this. This was the highest for some reason. So regarding like durable good expenditure, the vast majority of people spent, well, not the vast majority, the majority of people spent between $101 and $250. The majority of people spend less than that, and still a big chunk of people are spending $0 again, despite the fact that the household income in this area is apparently very high. And then, so for boat owners, we also asked how much did you spend in the last 12 months on like boat upkeep? The vast majority of people spend less than $1,000, and again, another like probably 15% of people about spent $0 on boat upkeep in the last 12 months. So yeah, again, it's interesting the fact that it's a high earning area, and the people who are responding are high earning, but they don't spend that much. And so now that we have the survey data, we could finally go into the next steps of the project. So obviously there's going to be a more detailed statistical analysis, regression analysis of the survey data. A big one is focus groups with volunteers who completed the surveys, so that we could get more anecdotal information or like data and whatnot that could not have been input into the survey that we might find valuable. And implant is probably the most interesting statistical part of it. Basically, we take our data, we put it into an input-output model, and it produces the data that we actually want. So basically, we could find, based on the data that we collected, on the individual expenditure and the amount of people and the frequencies, we could then theoretically find out the direct effects, the indirect effects, the supported jobs, and then we could also model how maybe down the line certain management choices will affect the state economy into the future. So conclusions, surveys and interviews are an effective avenue for collecting data and characterizing an area. Recreational marine fishing is an important contributor to local and national economies, supporting thousands of jobs, generating millions to billions in tax revenue, depending on the level of government. And managers must work more intimately with recreational fishers into the future to maintain fisheries that are both environmentally and economically sustainable. And that's my presentation.
Hello, everyone. So my name is Illusion, and I will be presenting the American lobster fishery and derelict lobster trap removal project that I've worked on this summer with Cornell Cooperative Extension of Suffolk County as a fisheries technician. So for a quick introduction, since 2010, CCE, or Cornell Cooperative Extension of Suffolk County's marine program, in collaboration with the lobster industry, local officials, scientists, and the community, they have removed a lot of derelict lobster pods or traps from the Long Island Sound. The removal of these pods has made it possible to release many of the trapped species, and also the removal of the pods has cleaned the Long Island Sound through basically taking out any marine debris and gear. So the gear that's usually retrieved from the Long Island Sound is collected and recycled or returned back to the owners if tagged. So the motivation for this project is the growing importance of protecting marine species and also to prevent pollution in our waters. So as we know, marine debris is in the forefront of pollution in our water. Additionally, the Southern New England stock of lobster population, and which includes the Long Island Sound lobster population, is in very poor condition, and they are in decline and have been in decline for years now. And this has also decreased the catch effort from many of the lobstermen, resulting in more abandoned gear, and also with storms and any just crazy weather, they are lost and they pose a large threat to other fishermen that can basically get their own gear tangled up and species in the Long Island Sound that get trapped. So jumping into some of the life histories of the American lobster, they are long-lived solitary crustaceans, and they range in the Atlantic coast from Labrador, Canada to North Carolina in the United States. They are considered to be territorial species, usually ranging from 5 to 10 square kilometers. Larger lobsters tend to migrate a lot more than small lobsters. Small lobsters prefer inshore waters in rockier areas. However, they do migrate to reproduce as well, and they prefer temperatures around 65 degrees Fahrenheit. Once females are mature, they can have from 5,000 to 10,000 eggs, and they become once they become fertilized, they become visible on the underside of the lobster. So there are currently 2 stocks in the fishery, one that makes up the lobsters in the Gulf of Maine and Georges Bank stock, which is healthy and increasing. They are not overfished nor subject to overfishing. However, the Southern New England stock, which makes up part of the Long Island Sound as well, the population in the Long Island Sound as well, is in poor condition and in decline. They have been overfished for years and are not subject to overfishing, however. Here I have a graph from ASMFC, which shows the abundance of the Southern New England stock relative to reference points. And the abundance of the lobster is shown by the blue line, and the blue line decreases under the threshold line, which is the red line, around 2003. So the American lobster fishery was the 2nd largest fishery in the region in the 1980s, and the Southern New England population or stock experienced collapse in 1999. And since then, large die-offs have also happened in 2002 and 2006. And this is mainly caused because of the major stressors of pesticide runoff, shell diseases, and high temperatures and other environmental conditions that have affected American lobster. I decided to also add this graph that just shows the landings by stock. So the blue line is the Gulf of Maine and Georges Bank stock, which has been shown to have a lot more landings because of the healthy population. And the red line shows the Southern New England stock, which has not been as healthy and successful, and the fishery isn't doing too well, so the landings are low. So the objectives of this project and the main goal is to retrieve as much derelict gear from the Long Island Sound while also preventing ghost fishing. Ghost fishing is a term used to essentially describe the animals that continue to get trapped or fished in gear that is no longer monitored or actively used by fishermen. So the big issue with this is because is basically that the fish that get trapped, or any organisms that get trapped for that matter, continue to rebait any of the traps that they're stuck in, attracting more animals and causing more deaths, essentially. But CCE has set a path for the long-term conservation effort to basically remove these traps and prevent ghost fishing while also restoring the natural waters and habitats of the Long Island Sound, which essentially gives the American lobster a better chance to survive and basically live out of the traps. So the approach taken, the first map at the top is showing the Long Island Sound study area, and the side scan sonar image on the left shows some of the pots that we believe are in that area, just shown by these like dots. I don't know if you guys can see it, the colors, but essentially that was used to figure out where these pots might be. And then we went out and got some of these. Well, this Cornell Cooperative Extension has been doing this since 2010, but specifically for this year so far on the 3 trips that I've gone on, we've focused on Oyster Bay, which is shown on the map to the right. And those are the 3 zones that we've visited so far this year. So for the methods, we use a grapple system to actually retrieve these traps. And the grapple system is basically grapples, 10 grapples along the line, and they're dragged on the bottom. Once the captain of the boat kind of feels that we have snagged some traps, we then use the hydraulic collar to bring these traps up on board and do the data collection of animals and the conditions of the traps. And later they're taken to the marina where they will be ready for pickup by the Department of Sanitation. So for the actual methods and data collection, we collect data on the trap conditions. So the percentage that they are buried, the condition, the damage basically done, the percentage of the damage done to the traps, and also the specific coordinates we have already visited and hauled through, and the species count, species length and weights, and any of the owners permit tags in order to contact them after the trip to see if they want it recycled or returned. So for the results of this project, so far 88 traps were removed from the Long Island Sound in 2023. And that's only in 5 of the trips. Today, actually, the team is out taking some trips. I believe they've taken out like around 20 today. So those will be added to the data set soon. And in those traps removed, we've seen 2 American lobsters that we accounted for and later released, which is actually some of the data is collected and displayed on the table below. But since 2010, there's been 21,931 traps retrieved from the Long Island Sound, with a total of 4,331 live lobsters retrieved as well and released. These are some results from the Long Island Sound study, and they're just showing the lobster abundance and lobster count per tow from 1984 to 2022, and it just shows the fall index and the spring index of lobster count per tow, which is very low and has been decreasing over the years. In 2022, the total traps retrieved were 1,046 traps, and the most common animals found were rock crabs, and the most common fish found were tautog and essentially blackfish, which were the most released, followed by black sea bass, butterfish, and the oyster toadfish. And this graph here shows the amount of live and dead animals recorded and removed from the traps throughout the 2022 project, with the total of 1,046 traps retrieved that year. Now we're still planning to do a lot more trips this year, so the 88 traps that have been retrieved are just for the 5 trips so far. So there's going to be a lot more trips in the future. So far in 2023, spider crabs and rock crabs were the most common, and the most common fish we saw were scup and black sea bass. So in conclusion, the American lobster stock, specifically the one in southern New England region, is continuously in decline, and this is most likely because of the increasing temperatures, which would continue to drive the population out of the Long Island Sound and continue to have them shifting northward. So it is important to take precautionary measures in order to prevent any of the mortality that traps might be posing to these American lobsters. And of course, the derelict lobster trap removal project is a huge solution and help to the American lobster population because of their continuous work to prevent ghost fishing, and the partnerships that have also been created over the timeline of this project has expanded environmental awareness and improved communication overall in order to basically improve the fishery. So now I just want to acknowledge everyone that helped me stay on track and basically supported me throughout this whole project. So Tara Ryder, my advisor, and the whole fisheries team from CCE, which have been very helpful and made my summer very fun. Here are some pictures of the trips and things I've done this summer as well. I've done some work with striped bass as well. I've done a lot of fishing this summer for the PCB contamination assessment. I went on a summer trawl as well for a couple of days. I've done monkfish marketing, and the summer trawl was for the squid fishery, which was very fun. I've never seen so many fish in my life. Yeah, that's it. Any questions?
How did you like market your products, like your work at shirt?
Oh yeah, so I actually did create a couple of social media platforms for well, I didn't create I created the accounts for a lot of social media platforms this summer for monkfish specifically, and for choose local fish. That reminds me, I have some surveys that you guys should definitely take in order to help us learn more about monkfish. I'm on the job right now. And yeah, so if you guys can take this whenever you can, I'll leave them there, and you guys could just leave them and I'll pick them up at the end. But yeah, thank you.
Thanks.
That works too.
Okay, hi everyone. My name is Jessica Salmon, and my project was on Jamaican special fisheries conservation areas, basically Jamaica's MPAs, effect on snapper and parrotfish species abundance and parrotfish species life phases. A not so simple story. So a little bit of background. Our parent project umbrella, I guess you could say, is the Global Finprint Project. And that's basically a global database of underwater footage that scientists around the globe can look into and use and see global trends. So it's a very important database that was created. And my subset, well, Stephen Heck and Brad Peterson back in 2016 and 17 went to Jamaica and set some of these underwater footage to collect, and my job was basically to review all of them, so that was very fun. And also the usage of this data set is to look at these marine protected areas in Jamaica, and to see the difference between inside and outside of a marine protected area. And as we all know, they are
they can function as no-take zones, which reduces the fishing pressure in this area. And they also produce a spillover effect for the fishermen that are in the area. So the fishermen actually support these, and they were involved in the legislation such as the fisheries industry regulations to put these in place. So again, Special Fishery Conservation Area is just an MPA in Jamaica. So the motivation for this project was the use of underwater footage. So this is fisheries independent survey method, which is a less invasive way of sampling the waters inside versus outside of a marine protected area. So like we heard of eDNA, which we didn't use in our lab, but basically my job was to watch hours upon hours of footage and identify everything and count everything. So it's a very long process. So I've been in this project for quite a while. But the goal is to see if there was a higher abundance, relative abundance within a marine protected area versus outside. And moving forward, we could potentially use spatial prediction models to best place these MPAs and also create better evaluation systems for these MPA efficiency. So Steve and Brad went to 5 different locations, but we're just looking at 2 because these were the 2 locations that had a good portion of the videos both inside versus outside of the protected areas. So then we could actually compare a little bit about the population densities. So you can see that Charadrius is on the north shore, which is a coastal area, whereas Pedro Bank is an offshore bank that is popular to a lot of species. And for my methods, we looked at these beta remote underwater footages. Just imagine a tripod with an arm with a bait cage out in front. So it tracks anything in the area, and you might say to yourself, okay, parrotfish aren't carnivores, how are they attracted to a bait cage? Well, they're very territorial fish, so of course they're going to see what's placed in their area. My measure in this project was the max N, so that was the maximum number of individuals in a camera shot of the video, so we review it for an hour. So the maximum number in the same species and life phase if we're talking about parrotfish. And then the average is just the average of the videos inside versus outside, or any of the comparable factors that we will talk about later. And those are just some definitions for you. And then significant difference was determined by 1-way, 2-way, and 3-way ANOVAs with the addition of a Tukey test. And that's just an example. So closer up, you can see that maybe you can't see the colors, but there's videos inside versus outside, and then you have the breakdown for the 2 locations. So we had 86 in total, with 43 in both location, which actually worked out pretty well. And this is our 3 functional groups. So in order to see the efficiency of the MPA or try to gauge, okay, is there a difference between inside versus outside? You have to look at multiple trophic levels. So we have the herbivores made up by the parrotfish. We have the yellow-tailed snapper, which are the invertebrores or planktivores. And then the rest of the snappers that were in that our target species, you could say, were the Lujanus snappers, which are carnivorous, they eat smaller fish. And the X's on the other side to show which location they were present in. So, and this is all the fish that I had to know. I also did this for everything else earlier in this project, but we won't get into that because that's not for this project. And this is an example of why we equated the life phases differently for the parrotfish, because this is all one species. But looking at them, you could never tell that. So they have different life histories, and that reflects why they have different, they look different, and they are still in the same place. So all 3 of these individuals could still be seen in the same location. So my objectives / my research questions, because I wrote a thesis for this and hopefully get it published. My 3 questions are: how do the SFCAs, MPAs, affect the abundance and the depth of these drop locations? Does that affect the abundance of these groups? And does the age of the marine protected area affect the abundance inside versus outside?
Now, so these are a lot of results. This is just my table of contents, but there's 5 different I don't know why they all became ones. Don't mind that. But you have 5 different locations. You have the functional group population density. So we have those 3 functional groups. We have the life phases for the parrotfish. So they were proportioned. So dividing by the number of species that were seen for each of the life phases. So we had 2 for juvenile, you can read, but I won't get into that. And then there's 4 species that we looked at individually by life phase for the 2 sites, because those were the ones with the highest frequency. The other species were only seen once or twice in the 86 videos, and it didn't make any sense to try to compare those. And then the 4th section was species-specific life phases, so just looking at the juveniles and the species, and then initial and terminal. And the last group was the depth and the age of the MPA and seeing if they changed the population density of the fish we were looking at. So in the first section, just the functional groups, so the 3 different functional groups, they all varied by site significantly. In the snapper groups, they were more popular at Ochos Rios, whereas Pedro Bank had a higher relative abundance. The parrotfish had a higher abundance at Pedro Bank, but none of them overall varied by MPA, which was quite interesting. And for the 2 groups that were made up by multiple species, they all varied by species. So for parrotfish overall at both sites, they had different dominating species. But for Lugana snapper, because the snapper were seen as frequently, it was only seen that at Ochos Rios, there was a difference between snapper. So Pedro Bank, the species weren't significantly different from each other. So for my second section, looking at the life phases overall, we saw a significant difference for the initial phase, which saw more initial phase parrotfish with all those species combined, inside versus outside, which is huge because this is significance here. But Pedro Bank did not show any significance other than just having more juveniles in the area than terminal phase parrotfish. And we have our species-specific trends for parrotfish. We have the red band here depicted in the graph. As you can see, there was more initial phase than terminal phase. For the princess parrotfish, you had a significant interaction between MPA and life phases. And that basically just means that at Ochos Rios, which was the site that drove this interaction, there was a possibility that there could have been more, could have been less inside of MPA. However, when we ran the ANOVA test, we could not, the value was not enough to separate those. So we can't determine that by significance. And at at Pedro Bank again, just like here, initial phase was greater than terminal phase, and also juvenile phase was greater than terminal. Yeah, so both of the other 2, so terminal phase was the least for princess parrotfish, and then striped parrotfish only differed by site. So there was more at Ochos Rios versus Pedro Bank, which is interesting enough because that does not follow the overall trend that we saw before for parrotfish. So striped parrotfish don't like what's common for, parrotfish. So and my last species that I looked at specifically was the spotlight parrotfish. All you have to see here is that there was more initials than terminals again. So I don't know what terminals we're doing. And that the MPA was marginally insignificant. So as you can see, they're trending towards inside the marine protected area versus outside. So that's, and then Pedro Bank was not significantly different at all for the species. And then, got it, 5 minutes left?
You're at 10 minutes.
Okay, cool. So species specific life phases. Basically, these are just looking at this life phases by themselves. So for juvenile overall, there was more princess parrotfish than spotlight parrotfish. And then for initial phase, overall there was at both sites there was a difference with the species, but also, as we saw before with the overall trend, that density varied by MPA. So there's more inside versus outside, which was also seen. And then also, as you can see on the graph depicted by the letters, is the species were different. So here we have a significantly greater amount of striped parrotfish And then also princess fish were quite significant as well. But at Pedro Bank, we have more red band parrotfish, which was interesting difference between the two sites. And then for the terminal phase, same thing like the initial phase, they varied by species. So here again, striped parrotfish dominated Otros Rios, and at Pedro Bank, red band parrotfish dominated. So that was quite interesting. And then our last section of results, which was looking at the depth and the age of marine protected area. So the only functional group that varied by depth was the yellow tail snapper, which they preferred a deeper location versus a more shallow location, which was interesting enough. And I just used the average because there wasn't a significant interaction between depth and site. So I could use the same depth for both of them. And then, like I said, there was no difference with the age of marine protected area. And the broader significance and impact was the benefit to marine conservation and policy for this project is it aims to increase collaboration, which was already stated. It's a non-invasive sampling technique, and it promotes the use of technology to solve conservation issues. The larger research question, in my opinion, is what is the best way to test how MPAs are actually affecting fish populations. And this was just an example of a crigging map that I made in ArcGIS Pro, which shows a hotspot for the red band parrotfish in Otros Rios, actually outside the marine protected area and further east than the marine protected areas. And these are my references, and I have a thank you video for someone in the audience would think that's pretty funny. Okay, you guys can ask questions.
Play.
Oh gosh. It doesn't have any sound. This is just Brad Peterson and Steve and them setting up the video to have them put over. So you can see the bait cage in front, and they just shove a bunch of fish in there to attract the fish. So.
It's just about your methods. I'm just curious, like, what was your approach to going through all those videos?
Oh, oh, good lord. Probably over 400 hours. I've started working on the rest of the videos this past year, so technically did 9 credits on this project, but yes, it was a lot of videos, and I started as an undergrad here, and I worked with the videos as well, but it was only a small subset. It was only 12 videos at each site, but it was 5 locations, so it was 60. So this one was 86. So yeah, it's.
Did you see anything really cool in the video that you want to tell us about?
The moray eels. I also saw a bull shark or a bunch of different sharks. They come up. I didn't have enough time, obviously, to put it in, but they basically fit the cage in their mouth and then realize they can't ingest it, so they have to like figure out how to get out of the situation. So yeah, it was good for me.
Hi everybody, my name is Madeline. This summer I worked with the Cornell Cooperative Extension in Southfield with their marine program. And my project focused on investigating ecosystem structure comparisons between eelgrass restoration sites and natural meadows to determine recovery status. So specifically looking at sediment structure in these meadows. So let's just dive right into it. So what is eelgrass? It is a type of seagrass, which means it's a true vascular plant. And this means that it has a fully established elaborate root system and has flowers as well. But it is unique in that it can grow fully submerged in a marine system. They typically grow in monotypic stands and in coastal shallow waters as well, in a mix of different substrates from sand, gravel, and silt and clay mixtures. They can grow up to 4 feet in length, and they also reproduce both sexually and asexually. You can see in the bottom picture here, the rhizome structure, that's how they expand asexually, and then flowers, of course, produce the seeds for sexual reproduction. So eelgrass is important because they are renowned as being ecosystem engineers, and they also serve as a biodiverse marine ecosystem for a lot of commercially and recreationally important species. The Magnuson-Stevens Act established them as essential fish habitat in 1996. And then
coming back to that ability for them to be an engineer, they're able to pull suspended sediments from the water column and put them into their root systems to kind of build up their own substrate. And this really transforms the sediment structure to be able to hold more fine sediments as well as higher organic content. And they also are very good at erosion control, kind of curbing storm surge, wave action, carbon sequestration, and they're also fun to dive in. So unfortunately, on Long Island, eelgrass, as of the 1930s anyway, about 90% of eelgrass meadows have been lost on Long Island, mainly due to wasting disease from a slime mold, and then of course, the human activity pressures like dredging, shellfishing, and the hardening of shorelines has also kind of hindered the growth of these eelgrass systems. So Cornell Cooperative Extension, their marine program, established their own marine meadows program, specifically to help restore native eelgrass meadows in Long Island. So they have a couple of objectives: to track health of existing meadows, to restore viable sites, to develop alternative sources for eelgrass planting material, and then also to experiment with innovative planting techniques. So they have planting techniques like rock planting, where you put a rock over viable shoots and hopefully they will take root. This is really useful for a rocky system where there's not really a lot of sand to root in. Buoy deployed seeding is another method which we helped out with this summer in Tiana Bay with, where you take flowering shoots in a mesh bag and you deploy them by a buoy, and they'll kind of seed the waters throughout the season. And then the burlap disc or tortilla method, where you weave in eelgrass shoots into a burlap material, and you plant that into the substrate with sand as an anchor, and eventually the burlap will degrade, but the shoots will take root and hopefully grow from there. So getting into my project, I wanted to focus on that engineer quality of the eelgrass and to see if the status of the sediment structure could be used to determine the recovery status of CCE restoration sites compared to natural eelgrass meadows. So there was three kind of levels of comparison I was looking at. Restored versus a natural counterpart, so a nearby natural meadow that could be used as a comparison. Vegetated and unvegetated portions of the meadows, as well as sheltered versus exposed meadows. So these would be meadows that, in terms of sheltered, have very little wave action versus high energy from constant wave disturbance. So overall, you would expect to see finer sediments and high organic content in the vegetated and the sheltered meadows. And then ideally, if the meadow is restored, it should have similar sediment structure to its natural counterpart. So I looked at 4 sites in Long Island. So the 2 here at the bottom, Sag Harbor Restored and Sag Harbor Natural, are in the Peconic Estuary. These represent the more sheltered meadows. And the Sag Harbor Restored site from CCE was established in, they started planting 2016, and it was planted pretty much right next to established natural meadow. So it just made sense to look at those 2 sites. Those are naturally sandy sites as well. And then for the other 2 sites, I looked at Gull Island. There's a restored site on the north shore of that island there, which planning for that started in 2009. And they had a recent planning in 2021 as well. And those shoots for that site were taken from a natural meadow at Orient Point, which is also on the map, which represents the natural meadow that I looked at as that kind of reference meadow. Those are both rockier sites. So sampling took place mid-July in the span of a couple of weeks. And in the field, we took samples on scuba. So we went down, we had 3 10 by 10 inch quadrats, which we put down randomly to look at percent coverage of eelgrass. We also took video footage with GoPro to look at growing conditions. And then, of course, the actual sediment cores themselves. We had 6 cores that we took per site, 3 of which were used for grain size analysis and 3 which were used for organic content analysis. So here's just a couple of pictures. You can see here that PVC pipe I have in my hands, that's the cores that we used. So going into the lab, for the grain size analysis, we use methods described in a study by Folk et al. 1974 and Wentworth 1922. So this was where we sieved the sediment cores through first a 2 millimeter sieve to separate gravel, and then through a 63 micrometer sieve to separate the sand. And then the remaining silt and clay fractions were suspended in a 1,000 meter graduated cylinder, and that sample was extracted by a pipette. And then the organic content analysis, those methods were based off of Mudrock and Magnite 1994. So this is where we took the sediments and placed them in pre-weighed crucibles and combusted them in muffle furnaces at 450 degrees Celsius for at least 4 hours. And the weights were taken before and after combustion. That weight difference represents the organic content that was burned off. So looking at the quadrat comparisons, we grouped these into categories of percentages. So the Sag Harbor natural site and the Orient Point natural site had similar percent coverage between 50 to 75%. Sag Harbor restored site actually had the lowest percent coverage with 25 to 75%. And then the Gull Island restored site surprisingly had the most at 75 to 100% coverage. And this kind of discrepancy in growth was also represented in the blade lengths as well, where the Gull Island site had observed to have the greatest biomass, with some of the blades reaching up to 55 inches. So getting into some of the grain size comparisons, the gravel and the sand comparisons aren't very surprising. Obviously, the Gull Island and Orient Point sites were rockier to start with, so those have higher gravel percentages, whereas not so much at Sag Harbor sites. And then the sand composition kind of shows a similar trend where the Sag Harbor sites were all over 99% composition for sand. In terms of looking at vegetated versus unvegetated, there was only one significant difference, and that was between the Orient Point site and its unvegetated nearby zone, where surprisingly there was actually more gravel in the natural meadow, which you would think it would be the opposite. But some more interesting things that come up are more so with the silt and clay composition comparisons and the organics. So here you can see that the Gull Island restored site actually had higher percentages of silt and clay compared to the other sites, specifically the unvegetated site nearby. And again, that's a very high wave action zone, so you would think that it would have a harder time holding on to that silt and clay comp. And we saw a similar trend with the organic composition, where the Gull Island site did in fact have the highest percentages of organics. This did have a significant difference based off of t-test with its unvegetated zone, where it was actually almost double that of its unvegetated. So it actually had, I believe the numbers was 0.9 to 1.4% organic material present. And again, kind of surprising, you would think that that trend would be more so with the Sag Harbor site. So it's showing that the Gull Island site was successful in being able to retain that finer sediment and organic content versus right outside, like right next door to it in the unvegetated zone, there's kind of like nothing there. So in conclusion, Sag Harbor may not have enough biomass built up yet to keep strong currents from stripping away fine sediments and the organics. While it is sheltered from wave action, there is those occasional strong currents that do come through. That being said, however, it does have a similar sediment composition to its natural counterpart in Sag Harbor. And both meadows are still able to provide sufficient habitat for a diverse range of organisms. And then for Gull Island, in terms of this study, it's a established and successful CCE restoration site, which is really exciting, and it exemplifies how successful restoration can help to kind of engineer a healthier ecosystem. So moving forward, some other variables to consider. It might be interesting to look at in faunal diversity and biomass, as well as bioturbation activities that might be impacting growth rates. And then of course, water quality is always important, like temperature and light penetration. CCE does work with light loggers to track this data currently, and that will be ongoing. For future study potential, CCE has mentioned that they will be doing 10 test planting sites in the South Shore Estuary next spring. So this could be a good opportunity to look at sediment composition beforehand and seeing how it changes within establishing eelgrass system there. And then of course, Gull Island will be an important site moving forward to determine what environmental conditions have allowed eelgrass to survive and thrive in a high wave action and rocky surroundings. And I think that's it. I just want to say thank you to Kim Manzo and Steve Schott, my CCE mentors. Jason with CCE really helped with dive coordination as well. My MCP colleague, Anna Meikenbaum, for project support, and then my SoMAS faculty advisor, Dr. Brett Peterson.
One question, I think.
Yes?
I might have missed it, but did you mention when the recovery project was actually done at Gull Island?
Yeah, so they did a first planning in 2009 with a follow-up monitoring in 2010. And then they actually didn't revisit the site until 2021, where they did a follow-up planting. Yeah, it's doing really good. So any other questions?
I think we've got a.
Okay.
Okay. So hello, everyone. My name is Eureka. My academic advisor was Professor Ellen, and Emma Rudazzo was my advisor throughout the whole project. And my study was uncovering the hidden anthropogenic debris, scientific study on collecting microplastics pollution in Shinnecock Bay. Okay, so this is my table of contents of what I'm going to be presenting today. Because of time, I'm just going to pass by this. So a little bit of history of Ponquag Bridge. It was first built in 1930, and it was the only way access from the Atlantic Ocean through Hampton Bays. In 1986, there was a construction of new Ponquag Bridge, which is this one right here. And then during Superstorm Sandy, closed off the old Ponquag Bridge because of its collapse, which is right over there. Today, there is a north side and south side. The south side beach is home to recreational activities such as fishing. There's a boat launch there, scuba diving. You can see there's a dive flag right there. Picnic tables and other things as well. And my project was on the south side of Ponquag Bridge. So a little bit first about Mission Blue. Mission Blue is an international recognition in protecting the ocean and marine life. They have things called hope spots, which is basically recognized through Mission Blue and further protecting these areas. They look beyond marine protected areas. So for example, marine protected areas in total, there's 12% of land that's being protected and 5% is protected through ocean. And currently there's 156 hope spots currently right now, and Shinnecock Bay became a hope spot in 2022. So as we know, plastic is everywhere. There is two main types. There's macro and microplastics. Macro is greater than 5 millimeters, and then micro is less than 5 millimeters. Macroplastics can come, of course, in various sizes and break down into the category of microplastics. And the impacts of them is the ecosystem environment, indigestion, toxicity, harmful chemicals, and damage of the organs. For example, like fish can by mistakenly eat microplastics, and that causes more mortality rates. So plastics ending up in the ocean. As we know that we produce around 38 million tons of plastic and being produced globally every year. 3% ends up in the ocean. And based on 2010 statistics, that was around 800 million tons. This diagram basically shows the sources, pathways, and receptors. So pathways can be through production and what we use it for, and then the pathways can be through land, through the air, or through the sea, and then what being end up in different types of environments, and ending up in different types of organisms. And this can directly affect them, but also indirectly affect things like the economy, like for tourism, for example, like if there's beaches that are full of plastic, that's going to decrease the tourism rates and overall basically collapse, which is not a good thing. So the materials that I use data collection equipment. This is a debris dive is what we called it. We have 3 of them for each zone. I'll explain that more later. We have, of course, scuba diving equipment and also different types of utensils and specialized equipment that was used during a dive, such as a dive bag. There's sometimes too much debris inside the bag itself that's too heavy for one person to carry. So we have dive bags to make it easier to carry through the water. So Shinnecock, this is basically Shinnecock Bay. The inlet's right here. New and Old Pong Bridge is right here. South part of the Old Pong is over here, right along June Road. And that's the west side and east side of Shinnecock Bay. To zoom in a little bit further, I broke it down into 3 zones. The first zone was 0 to 6 feet. Second zone was 7 to 13, and third zone was 14 to 20 feet. How we communicate that underwater, we have depth gauges that we see through our computers. We also do different signs, like this is first zone, second zone, third zone. So that's how we did that underwater. How I basically broken down the categories for different types of materials, not only limiting plastics. There is this thing called PADI Wear. PADI itself is a scuba diving training agency, and they have this thing called Dive Against the Breeze, which is a conservation platform. This is how it looks like on the map, and then these little circles are just different types of sites that people have uploaded their information and data through different dives that they have done. Their goal is to reduce marine debris by 50% in targeted countries by 2030. This picture on yours, whatever side it is a slate. Basically, it has different types of materials which are categorized in plastics, glass, ceramic, metal, rubber, wood, cloth, paper, cardboard, and other mixed materials. So this is really helpful to break down and tally up at the end for each dive. There is a website version of it online as well. So there was a total of 20 dives throughout basically the whole months of summer of June, July, and August. I have the dive date, the high tide, dive time itself, moon cycle, and the visibility. You can see here in June we had around 15 foot of visibility, and then it drops down to some days were 5, some days were a little better, but this was basically because of the algae that's blooming basically during the later months when it gets warmer, that causes the limitation of visibility. So I was going to ask you this question, but I was going to say how much total weight did I find? But the total is 675.2 52 pounds. That was the total for all 20 dives. This graph basically shows all that together. The numbers on the top is color coded with it based on zone. And the number on the top basically shows the highest amount of weight in that zone during that dive. There's no significance in terms of which zone was more favorable than the other in terms of weight. But also we have to think about the dive time, visibility, and how much debris is in each zone that affects how much is being taken out. So for example, there might be a day when it's very limited visibility. So we might stay in one zone versus another zone, more shallower, for example, and spend more time in that area compared to others. So for zone 1, this is only for all the macroplastics, not the other types of materials. In zone 1, there are a variety of different types of materials in terms of plastic. If you look closer to specific types, there are much lighter plastics than heavier plastics, and there's a more variety of it. for zone 1, which is the shallowest zone. And then going to zone 2, we start seeing a little bit more of fishing line and rope. So there's 55.6 of fishing line there, and as well as 22.9% of rope in that zone. And also for zone 3, we see that there is 74.1% of fishing line in zone 3, and then 17% of rope. So for zone 2 and 3, this indicates that there are hotspots. Hotspots for fishing, which also can cause for entanglement, which is going to be for later. So trends and significance. So this graph shows the comparison between monofilament fishing line versus just other plastics in general. We can see that there's no correlation for just plastics with no fishing line. But you can see for zones 2 and 3 for fishing line, that there's a lot more compared to the others, showing that these are indicators for hotspots for zone 2 and 3, which is also under the right where the bridge starts and off the fishing pier. So for entanglement, when there's fishing line, there's also going to be entanglement. So spider crabs have the highest amount of entanglement rate, and then there's horseshoe crab, green crabs, and sea urchins. You can see in the picture over here that basically shows, it's hard to see in the red, but where the entanglement is, and some of them are in the joints. For the horseshoe crab is right where the tail is. The reason why that spider crabs might have more entanglement compared to others is, one, their population. There's a lot more population in that area, as well as they're more fidgety compared to other crustaceans. So a whole summary of my project was that there's a variety of debris ending up old Pogpog Bridge. They vary in size and weight. And there's also marine life going in on the debris, which kind of have this ethic questions of, is it right to take out debris even though it has become kind of like an artificial reef for maybe like tropicals that come in through the summer. There was a study that talked about this, and their conclusion was no one size fits all, which basically means that even though it might be an artificial reef, it can be beneficial, but it cannot be beneficial. So for the fishing line, for example, there were like different sponges growing on top of the fishing line, but because there's a lot of entanglement, it doesn't really benefit in that kind of environment. There were things like the mixed material foundation that I found over there in zone 2. There was a lot of things going on it, and because there wasn't any entanglement on it, and it didn't seem like it would harm the bridge or the habitat, we did keep it after we weighed the material. So additional studies that I would like to do maybe in the future is doing comparative analysis between high and low tides. This would be especially good for zone 1 because it's being exposed from the water, so you can probably see more of what at the surface. Another study is debris found at New Pongquag compared to Old Pongquag. I did recently go to New Pongquag Bridge, and I did find a lot of lines again, also different types of traps, fishing traps, which is really cool to look at, but also like we should probably take those out, and also a boat ladder. That was also cool, but I couldn't take it out because I did not have my lift bag. And then also North Shore versus South Shore debris, also doing kind of like comparing analysis with that. So what's next? How can you get involved? So I did my project through Hampton Dive Center. They have this diving club called Aquanux. We also have this thing called Dive Against the Breeze, which is from Patty. We have our next date on August 26th, so people just come around, make sure you do have like a parking pass if you do come and join, because Pongquag does have a parking pass for that. We go there, make sure you bring like debris bags, and then at the end we just like tally everything up, and then we could post it on Patty Aware, which would be nice for people globally to look at. If you're not into scuba diving, or you don't have a certification for scuba diving, Relic also has their thing called Protect Our Oceans, which is basically a basket, and you could just pick up trash at the surface, which is pretty good as well, and also try to get involved. And that's it. Any questions? Yes.
Yeah, I was just wondering, I mean, I did the math, I thought about 70 pounds.
Yeah.
That's like 32 pounds per day.
Yeah, so per day, of course, it's not every day, it's around 33 pounds. There might be some days that I might, like, for example, like visibility, or if we have like some kind of unfortunate emergency, like we might not be able to go out as far as we can. Current can also be an issue. Sometimes high slack tide, depending on the moon cycle as well, varies. So sometimes like 15 minutes, other time it could be a lot lower or higher than that. So that also takes into account of how much we do get for each day. But yeah, it's a lot of debris.
Like, how is the bag streamlined to help you carry all that?
How is it streamlined?
Yeah, you said it was like.
So what I do when it's very heavy, I just hug it like a big pillow and just like inflate my VCD so I don't like crawl against all the pilings and all that. So, yep.
Great job, very interesting. I'm in Ellen's lab, we're in Bay all the time, and I didn't realize there was so much debris around the area. We have been contemplating putting up signs, like to promote the Hope Spot around populated visitation areas, and this is an important component for fishermen or fisher people who go out recreationally, just to be conscious of not littering with their lines and all that. But I would so thank you for bringing the issue,
to more awareness.
forward.
Yeah.
I was wondering whether any of the spikes in late July that you showed, I didn't, memorize the data there, but it looked like there were some spikes in when there were highest levels of debris. Do you think it could be related to visitation?
It can be probably because why it starts spiking up. Probably be good because it's warmer weather, people are starting to come out more. Another reason why is because how I took for June is kind of what worked in the in my practice and what didn't work, and so there's some changes that I did need to make. But overall for that chunk, even though there was limited visibility, we started seeing different types of patterns around the area, and kind of we know we have to go this part or this part or this part before exploring it even further. So even after 20 dives, there's still a lot of things that I found that I couldn't get to pick up because it was too heavy.
Right.
And there was
zone 1 is the shallow zone.
Yes.
So if you look at July 27, 29.
Yeah. So
30, that could be litter.
Yeah, it is.
People going to the parking lot and it's blowing in the water.
Yeah, so right before the road bridge starts, there's a trash can that's there. One, it's always filled up or overfilled, and two, of course, there's the seagulls that probably pick it up and then just throw it all over the place. There's things like that that can cause it, either man-made or just other things. Yeah, not fun.
Thank you.
Let me grab my notes. All right, so my name is George. My internship was I was writing up a 5-year review on the common angel shark, pictured in the background. My faculty mentor was Carl Zafina, and my internship supervisor was Celeste Stout, who was an MCP. We've had a couple MCP graduates. I was listening, I think I was earlier in our seminar, but we have another one who I was working under for this internship. And quick disclaimer, I saw so many interesting photos of people doing field work. My internship was 100% remote, so I will not. I didn't feel like it made sense to have a picture of me behind a desk. So let's go. Let's move on. So the internship took place with NOAA. I'm assuming everyone here is familiar with NOAA, but I'll give a quick background slide about them. Their mission statement on their website was that NOAA Fisheries is responsible for the stewardship of the nation's ocean resources and their habitat. They provide vital services backed by sound science. And specifically, we're going to look at, if you look at the end of that quote on the second slide, they talk about conservation of protected resources and healthy ecosystems. That's where my internship really took place. So, quick internship background continued. My work occurred within the Endangered Species Conservation Division. There's like a million umbrellas, like there's a million divisions and umbrella divisions, and it was, so even doing this presentation, I forget what falls under which umbrella, but it took place within the Endangered Species Conservation Division within the National Marine Fisheries Service Office of Protected Resources. My group was specifically PR3. The Endangered Species Conservation Division's responsibilities included Endangered Species Act listings. And as you can see, and if you are lucky enough to ever join in on a meeting with NOAA this year, they have that little ESA 50, because it is the 50th year anniversary. They have that behind every single one of their backgrounds. And they didn't give me one, but they all had that as their backgrounds on the Zooms. So Congress enacted the Endangered Species Act to provide a means whereby the ecosystems upon which endangered species and threatened species may be conserved, and provide a program for the conservation of such endangered species and threatened species. And that's where my work really comes into play. So let me just move my name out of the way so I can see my slide. So through the Endangered Species Act consultation process, NOAA Fisheries works with agencies to design and implement actions to help promote their survival. The division provides leadership regarding listing decisions, development of rules, critical habitat designations, all of that. And yeah, I guess that last bullet point really just, it really just hammers home the leadership that they provide regarding rebuilding species abundance and controlling threats. And that's kind of what my division really took. Really, that's the kind of the matters they took into their own hands. Oh, there we go. So a little bit of more background about the 5-year review process that I undertook. So NOAA is required at once every 5 years to assess the current listing status of a species and make sure it remains accurate. And there's no specific time frame for completing a 5-year review, but once it has been initiated, it can't be unreasonably delayed. And it was delayed to a point where it started the review for the common angel shark started in the year 2021, but there was such a backlog, and just the existing PR3 members really didn't have the time to do it. So that's where I was lucky enough and fortunate enough to kind of come in and contribute to that. So the one sec. Oh, here we go. A 5-year review is a periodic analysis of a species status to ensure that the listing classification of a species as threatened or endangered is correct. And it occurs over 5 steps, a 5-year review. The first one is gathering information. The second is completing the template, which we'll look at that template a little more later, which concludes with synthesis, results, recommendations. The last few steps, 3, 4, and 5, are kind of procedural. They're peer review, concurrence process, and a notification of results. Steps 3, 4, and 5, I don't really undertake as much. Those are just kind of what happens when the 5-year review that I partake in is complete. So, information gathering. As soon as a species is listed on the IUCN Red List, all researching, monitoring, management that really comes into effect and implementation programs begin. And new information becomes available. Appropriate conservation efforts are hoped to be put into place. And they want to have the best scientific information being put in place and helping their efforts. And a 5-year review gives NOAA the opportunity to look at all the best information and kind of centralize that information. And it's important because in addition to just reviewing the classification of a species, it helps track the progress towards recovery. It can suggest appropriate next steps for its conservation. It can help with funding allocation decisions, help regarding permitting decisions, help determining whether to update a recovery plan, things like that. Yep. So kind of talking a little bit more about my information gathering stage. At the beginning of the internship, the NOAA library was very helpful in kind of gathering all new information over the last 5 years. They took information from resources like Google Scholar and Web of Science. And I know if there's any biological oceanography classmates in the meeting room right now, they all know about Web of Science and how fantastic it is. And I undertook some of my own research on that as well. The literature includes journal articles, academic articles, interviews with state, other experts. And with the documents we were looking for were anything that could help provide with biology, as you can see in the last bullet point. Biology includes distribution, abundance, demographics, genetics, all information like that regarding the common angel shark. I was looking to put into the 5-year review. We also want to find conservation measures that benefited the species, like threat status and trends as well. So gathering information was about a 2-week-long process, and we weren't just looking for any new information that we could find. For example, I'll give you an example from my internship. I found a new leech that was for the first time documented on the common angel shark in a paper, but because that had no information towards their status, I was kind of told to be disregarded by my advisor. It's an example of information we didn't want. So this was something I undertook recently, which was kind of listing the threats under a 5-factor analysis. The 5 factors are listed as here, but I'll read them out loud as well. 5 important factors to look for: present or threatened destruction of its habitat, overutilization for commercial, recreational, scientific, or educational purposes, disease or predation. For example, talking about the kind of leech we were talking about earlier, that had no effect on that showed no negative effect toward it. So that's why we didn't include it toward disease or predation. Just an example. Talking about inadequacies of already existing regulatory mechanisms and other natural and man-made factors. Not much applied to that last category in my report, but those are the 5 factors that are listed in the template. So once we finish it, what's next? There are 4 possible recommendations to make. You can uplist the species and reclassify them. You can downlist them. You can remove the species from the list entirely, or you can maintain the species' current classification. For what I'm doing right now in my internship, and it's actually, I think, funny in that at the conclusion of this, I have a meeting with Celeste to discuss my synthesis, and I'm writing out my result right now. But spoiler alert, when the 5-year review comes out, the common angel shark status will not have improved. It will remain critically endangered. The decline is attributed to just demersal fisheries, bottom trawling, really hindering the species. You specifically see it in the Mediterranean. There is the Canary Islands, which have shown a rich and diverse environment for the common angel shark, and it has the proper environmental conditions necessary for its life history stages. There's a productive marine environment. Like I said, there was a ban on trawl fishing in 1986 in that area, and that's kind of a little, I would say, hotspot for angel shark populations. And I only had 15 minutes, so I didn't want to get too into what I wrote specifically in my report, but that is touched on in there and kind of is a beacon for their population. Regarding peer review, sorry, let me go back one. I missed one thing. I also talk about, I give, I want to say advice. I give recommendations for different regulatory measures that can be put into place. I know it's for not just angel shark, but switching from J hooks to circular hooks are much better, and that can help. Importance of education of fishermen is something that has been documented to have success. So things like that will be included in my results and synthesis and future recommendations. and this is kind of where we get into procedural stuff. I know I only have, I'm running a little bit over time, so I'll try and make this quick. In the case of the Common Angel Shark 5-year review, all previous literature was peer reviewed, and there are different factors determining whether a 5-year review typically results in a peer review. But for my specific scenario, because the status is unchanged and there's not much new information based off of all the information NOAA has given me, I would believe that the paper will not undergo a peer review. The next step is a concurrence process. For Fish and Wildlife Service, the field supervisor must approve. That stage has not happened yet currently in my 5-year review, but we are getting to that soon. Written concurrence must be obtained from other regional directors in the regions which the species occurs. And I just realized, I don't think I gave an exact area where they occur. They occur in Europe, coast on the kind of that northern Africa, Mediterranean Sea. You really see a lot of angel shark, but mostly the Canary Islands is that real hot spot off the coast of Portugal, in the middle of Portugal and Africa, in those waters. So the final step in the 5-year review process is the reporting and public notification of results. And that can occur in a few ways. The first is a posting of a 5-year review on regional and national websites. Another way is the inclusion in the biennial report to Congress. And there's a third and optional way, which is an announcement in the Federal Register. And these are all mostly just procedural things that are just getting the reports out, making sure they're common knowledge to anyone who requires them. If you want to look at 5-year reports for any species, you can find them on NOAA's website. They were really useful to me when writing my report. So if anyone's further interested in what it looks like, I would point to that as a source. And kind of just to sum up my work with NOAA, I'll give another 1 minute. I thought working with Celeste, I discussed earlier, an MCP graduate, was very helpful, and she really understood where my situation, where I was coming from. She knew all the professors, which I thought was interesting. And right now, as I'm finishing my report, the angel shark that you see is not a common angel shark. That is a smoothback angel shark. And that is what I will be contributing to next when I'm done with the conclusion of my current report. And this is my works cited. Let me unshare my screen. There we go.
Okay, George. Unfortunately, you used all your time. And so these are concluding remarks. First, I want to congratulate all of the MCP graduating students. Good job, everyone. Please stay in touch with us. Let us know what you're doing next. Maybe you'll end up being a mentor for another MCP student down the line. Hopefully you'll all have careers that you're really happy with, and that this program will have made a difference to you in that regard. I also want to thank all of the mentors and faculty advisors. Without your help, this program really couldn't exist, and we could not offer this experience to our students. So thank you.